Ida say teacher one gala thar jahare hitila sigura
Good morning, PGTs in botany and zoology, that's in biology of all the EMRS schools. Now today, as we have decided that there is PGT in biology in all the EMRS, that is why we are involving both the PGTs in botany as well as PGTs in zoology for this program in the biology program. Now today is a term because yesterday was a PGT in where our research persons was a retired professor of botany. Today, sir, Dr. Prakash Paricha, is a very senior reader in RD Women's University, and he will be mostly dealing on human reproduction. So I also request all the PGTs in botany to take active role, because in your schools, your PGT in biology is there. So I know there may be some lacunae in case of the PGTs in botany, but as you are appointed as PGT in biology, so the botany staffs should also take some role. Now, I will give 1 or 2 minutes time, and I will request Dr. Paricha to start his deliberation. Just Dr. Paricha, please. 1 or 2 minutes of adjustment.
नहीं किछ नहीं सर
चाय मंगेबी नहीं
नहीं सर अच्छा मोर से डायबिटीज वाली जैसे इंजन
अच्छा सर डायबिटीज तो रॉयल लोगो यस स्टार्टिंग सर देखिए लूंगी सर
So good morning, teacher friends. Today we will be discussing multiple choice questions from human reproduction, which is there in Unit 6, Chapter 3. So in this chapter, you have both male and female reproductive systems. Then gametogenesis, fertilization, implantation. Then you have placenta formation. Also, the unit includes parturition and also lactation. The main purpose of this is to discuss multiple choice questions from this chapter. But prior to that, we will have a quick look at the contents of this unit. So, in this chapter, first of all, you have a male reproductive system. As you can see on your screen, the diagram shows the sagittal section, where you can see the structures of male reproductive system. It includes the gametes, it includes the gonads, that is the testes, and the ducts, sperm-carrying ducts, and also the associated glands, together with the male copulatory organ. As you can see in the diagram, the male gonads or the testes, they are located in a skin pouch called scrotum outside the pelvic cavity. Then on the posterior aspect of testis, you can see that bluish part that represents the epididymis. Then epididymis, it joins a duct called vas deferens. The vas deferens, it coils over the urinary bladder. Then it joins the ejaculatory duct. The ejaculatory duct then unites with ducts of bladder. Then it runs through the male copulatory organ as urethra. The diagram also shows the accessory glands which are associated with the male reproductive system. The glands you can see in the diagram that the seminal vesicles, prostate gland, and vulvourethral gland or the Cowper's gland. This is another diagram which shows the posterior view of male accessory organs of reproduction. Accessory organs, these are not concerned with the production of gametes, but they are part of the reproductive system. Here in this diagram, you can most clearly see the glands associated with male reproductive system, namely the seminal vesicles, prostate, and the vulvourethral gland or the Cowper's glands. Now you see a sagittal section of testis, within which you can see the seminiferous tubules. As you can see in the diagram, the testis is suspended in the scrotum with the help of a spermatic cord. The spermatic cord, it is externally covered by connective tissue. It contains blood vessels, lymph vessels, and some nerves. The testis, it has two coverings, as you can see in the diagram. Tunica vaginalis and tunica albuginea. The inner one is the tunica albuginea, and it projects at intervals into the interior part of the testis, dividing the lumen of the testis into a number of chambers. So you can see in the diagram a number of chambers. Each chamber is known as a lobule. In each lobule, you will see 2 to 3 highly coiled tubules. These tubules are the seminiferous tubules. It is actually within the seminiferous tubules that the sperms are produced. The sperm which are produced in the seminiferous tubules, they are carried into a network of tubules called retitestis, and the tubules which connect the seminiferous tubules with retitestis are known as straight tubules. Then from the retitestis through efferent ducts or vasa efferentia, the sperms are carried to the epididymis. Epididymis is located at the posterior aspect of the testes. The epididymis, it also contains coiled tubule through which the sperm passes. Then from the epididymis, sperm is carried into the pelvic cavity with the help of the ductus deferens. Now what you see is the cross section of testis. Testis in cross section looks like this when viewed under light microscope. So this is the microscopic structure of cross section of testis. You can see the cut sections of seminiferous tubules. Each seminiferous tubule, it is lined by basement membrane, then there is germinal epithelial layer, and in that germinal epithelial layer, the cells which produce the sperm, they are present. They are the germinal cells or the spermatogonia. Then the germinal epithelium also contains some non-germinal cells. They are the Sertoli cells. It is the spermatogonia which give rise to sperm, and the Sertoli cells, they have a supportive role. Within the seminiferous tubule, you can see the sperm in different stages of development. The development of sperm occurs with attachment to the Sertoli cells. The Sertoli cells provide nourishment to all the developing stages of sperm. The Sertoli cells also, they regulate the movement of spermatogenic cells. They also regulate the release of the sperm from the Sertoli cells. Sertoli cells also secrete inhibin. Then this diagram shows the male copulatory organ. To the left, what you see is the frontal section of the male copulatory organ, the penis. And to the right, at B, you see the transverse section of that male copulatory organ. You can see the male copulatory organ, it carries the urethra, that is the common passageway for sperm carrying semen and also for urine. Note here, the penis contains 3 cylinders of erectile tissue. In the transverse section, you can see the part which is labeled as corpora cavernosa. These are the 2 dorsal cylinders of erectile tissue. Then on the ventral aspect of the penis, you have another cylinder of erectile tissue, that is corpus spongiosum. These erectile tissues, when filled with blood, the penis becomes erected and is ready for intercourse. Then what you see is the sagittal section of the pelvic region of human female, showing the parts of female reproductive system. In this diagram, you can see the female gonads, that is ovary, then the fallopian tube or the uterine tube or oviduct connected to uterus. Also, you see the vagina. Also in the diagram, these two skin folds, labia majora and labia minora, they have been shown. This is a section of human ovary, which shows the internal structure. As you can see, externally, we have germinal epithelium. Then you see the ovarian cortex, and the central part is the ovarian medulla. The cells of the germinal epithelium, they give rise to the ova. In the reproductive period of human female, you can see in the section of ovary, the ovarian follicles containing the developing oocytes at different stages of development, starting from primordial follicle up to the mature follicle, that is the Graafian follicle. Initially, it is the primordial follicle. Then as it grows further, it becomes a primary follicle. Then it is converted into secondary follicle. Then ultimately it becomes a mature follicle. A mature follicle is known as Graafian follicle. It contains secondary oocyte surrounded by zona pellucida, corona radiata. It has a fluid filled cavity, that is antrum, filled with antral fluid. It has also a basal membrane, outer to which there will be theca. The ovarian follicles, they start their development in the cortical region of the ovary. As their development progresses, they become bigger and bigger. They move towards the interior of the ovary. Then ultimately, as the ovarian follicle, it matures, again it comes to the surface for release. In the diagram towards the right side, you can see a mature follicle is releasing its oocyte. Mark here, the oocyte is being released at a secondary oocyte stage. When the egg is released at secondary oocyte stage, it is surrounded by zona pellucida, a glycoprotein layer, together with the granulosa cells which form the corona radiata. After the release of egg, which is known as ovulation, the rest of the follicle is converted into an endocrine structure that is known as corpus luteum. If the released egg is fertilized, Then the corpus luteum is maintained beyond a period of 2 weeks. Otherwise, the corpus luteum will degenerate and it will become a scar tissue that is known as corpus albicans. This diagram shows the posterior view of uterus and associated structure. Here you can clearly see the two ovaries. Then you see the uterine tube. Uterine tube, it is attached to the uterus. A section of uterus has been cut open to show the internal layer. The cavity is also shown in the diagram, the uterine cavity. The wall of the uterus, it is thick, having 3 layers. Starting from inside, we have endometrium, then the middle muscle layer, that is myometrium, and the outermost, it is the perimetrium. The uterus, it has a cervical canal towards the inferior aspect, which opens into vagina through external os. And this diagram shows the external genitals of human female. External genitals of female are collectively known as vulva. It includes mons pubis, a mound of fatty tissue in the pelvic region, then clitoris, 2 pairs of skin folds, labia minora, labia majora. Also in the diagram, you see external urethral orifice and the vaginal orifice. The accessory glands of female reproductive system are the paraurethral glands or the Skene's glands. The Skene's glands, the paraurethral glands, they open on either side of the external urethral opening, and they secrete mucus. They are located in the wall of the urethra. Then other accessory glands of the female reproductive system are the greater vestibular glands, alternatively known as Bartholin glands. Located on either side of the vaginal orifice, and they open into the groove between hymen and labia minora. These glands, the Bartholin glands, they secrete mucus that helps in lubrication during sexual intercourse. Then what you see is the process of gametogenesis in male. That is the formation of sperms or spermatogenesis. The diagram shows the entire process of spermatogenesis in human male. The sperm mother cells or spermatogonia, they give rise to the sperms. The spermatogonia, they do not originate in the gonad. They have their origin during embryonic development, they have their origin in the yolk sac. Then the origin as primordial germ cells. The primordial germ cells then enter the gonads, where they develop into spermatogonia. So within the testis, we have these spermatogonia, which will give rise to the sperms. These spermatogonia, they act as stem cells and they keep on dividing by mitosis, producing more of those spermatogonia. During formation of spawns, the spermatogonium undergoes meiotic division. Spermatogonium, it is converted into primary spermatocyte. That primary spermatocyte undergoes meiotic division. It is a diploid cell. When it divides, it undergoes first meiotic division. It forms two cells. These cells are known as secondary spermatocytes. So during the formation of spawns, the secondary spermatocytes are the first haploid cells formed. Primary spermatocyte undergoes meiotic division to produce secondary spermatocytes. Then the secondary spermatocytes, they undergo second meiotic division to produce spermatids. So from one secondary spermatocyte, we can have 4 spermatids. Spermatids, they are haploid in nature, and the spermatids, they undergo transformation and form the spermatozoa. Here, notice one thing in the diagram that when the primary spermatocyte is dividing, it is forming secondary spermatocytes. And you can see the two secondary spermatocytes forms, they have a cytoplasmic link. They are not completely separated. Then also, as the secondary spermatocytes divide, forming spermatids, you can see the 4 spermatids, they are connected through cytoplasmic bridge. The cytoplasmic bridge ensures that there is a continuity between the neighboring spermatids. Some of these sperms, they will be carrying X chromosome and other will be Y chromosome. The Y chromosome is smaller in size, it is deficient in some genes, whereas X chromosome is bigger in size and it carries more number of genes. The X chromosome, it might be carrying certain genes which are essential for spermatogenesis. Those genes will not be there on the Y chromosome. So the gene products of those which are located on the X chromosome, they may be essential for the formation of the spawns. And through this cytoplasmic bridge, the spermatids containing Y chromosome, they can also have those gene products which are essential for spermatogenesis. That's why there is cytoplasmic bridge and the spermatids, they are not completely separated from one another. Then to the right of your screen, you can see the structure of a spermatozoon. It is an elongated structure having a head, middle piece, and the tail. Head contains the genetic material, the most important part, that is the nucleus. In front of the nucleus, there is a helmet-like structure, that is acrosome. Acrosome contains enzymes that help in penetration of the sperm into the egg for fertilization. Then the middle piece of the sperm, it contains mitochondria, which provide energy for the motility of sperm, and the tail. By vibration of the tail, the sperm can swim. Then this diagram shows the process of oogenesis. Starts with oogonia. As in males, oogonia, in females, they have their origin in yolk sac during embryonic development as primordial germ cells. Then they enter the female gonads, where they develop into oogonia. During a process of oogenesis, oogonium is converted into primary oocyte. The primary oocyte undergoes meiotic division, and it forms two cells, secondary oocyte and first polar body. First polar body is much smaller in size. This is the secondary oocyte, which will give rise to the functional egg. The primary oocyte, after entering meiosis, the development is arrested. Development is arrested till attainment of puberty. Then on attainment of puberty, the primary oocyte, it completes the first meiotic division, and it will give rise to the secondary oocyte and first polar body. The first polar body may subsequently divide, forming two more polar bodies. The secondary oocyte, at this stage, ovulation occurs. The secondary oocyte, on being fertilized, is converted into a functional ovum. These are the stages in the growth of ovarian follicle. It starts as a primordial follicle. The primordial follicle contains one primary oocyte surrounded by follicular cells. Outer to follicular cell, you can see the basement membrane. Then this one. To the right, primary follicle, as it grows, it becomes converted into primary follicle. Primary follicle, it also contains a primary oocyte. Then the follicular cells, they have multiplied. They are arranged in several layers around the primary oocyte. The cells which are arranged in several layers around the primary oocyte, they are known as granulosa cells. In between the primary oocyte and granulosa cells, you can see a non-cellular layer, that is the zona pellucida. Outer to zona pellucida is the basement membrane, and outer to basement membrane is the theca. As it grows further, it becomes converted into secondary follicle. Secondary follicle, as you can see, it contains a primary follicle. Here in the secondary follicle, you may notice a formation of a cavity, the antrum. Then finally, it is converted into a mature follicle or Graafian follicle. The Graafian follicle, wrongly it is labeled as primary oocyte. It contains a secondary oocyte surrounded by a glycoprotein zona pellucida. Then the granulosa cells, which form the corona radiata. A large antrum is there, filled with the follicular fluid. Basement membrane, you can see. Outer to basement membrane, we have the theca. There are two layers, theca interna and theca externa. The cells of the theca externa, together with the granulosa cells, they secrete estrogen. Then this diagram shows the menstrual cycle, the events during menstrual cycle, events which occur in uterus and also in ovary. As you know, the menstrual cycle, it is the length of the cycle is about 28 days, and it is divisible into different phases, starting with first menstruation phase, then proliferative phase, or the pre-ovulatory phase, then ovulation, and finally post-ovulatory phase. The first 5 days of menstrual cycle is the menstruation phase, during which the endometrium, the superficial layers of the endometrium, they are shed. So there will be bleeding, loss of blood together with mucus, certain tissue debris. It lasts for about 5 days, following which there is the proliferative phase starting from day 6 up to day 13. Then on day 14, ovulation occurs. The ovum is, the secondary oocyte is released from the Graafian follicle. Then the post-ovulatory phase, during which further thickening of the endometrium occurs. Side by side, you may notice the changes which are occurring in the ovary. Under the influence of the hormones, follicle stimulating hormone and luteinizing hormone, which are secreted from the anterior pituitary, changes occur in the ovary. The secretion of the FSH and the luteinizing hormone is under the control of hypothalamus hormone, gonadotropic releasing hormone. The FSH, which is secreted, the follicle stimulating hormone secreted from the pituitary, it stimulates the development of the ovarian follicles. As you can see in the diagram, the follicles, they are growing under the influence of the follicle stimulating hormone. The follicles, as they grow, they start secreting estrogen. Then ovulation occurs on the 14th day. After ovulation, you can see the rest of the follicle is converted into corpus luteum, which starts secreting progesterone and estrogen. Then look at the bottom part of the diagram. There it shows the levels of different hormones during the menstrual cycle. Levels of 4 different hormones have been shown: follicle stimulating hormone and luteinizing hormone, which are secreted from pituitary, and also the levels of estrogen and progesterone. Mark here, there is a sharp rise in LH concentration just prior to ovulation. The surge in LH, it stimulates the release of the secondary oocyte from the ovary. Then you mark a surge in progesterone in the post-ovulatory period. Then the level of estrogen, peak level of estrogen is found just prior to the ovulation. Then this diagram shows fertilization, the process of fertilization. When the secondary oocyte is released from the ovary, it enters the uterine tube or the fallopian tube, where fertilization occurs. This diagram shows a sperm cell penetrating a secondary oocyte. You can see the oocyte is surrounded by zona pellucida, and also you can see the cells of the corona radiata. With the help of the hydrolyzing enzymes in the sperm head, the sperm can penetrate the corona radiata and the zona pellucida to establish contact with the cell membrane of the secondary oocyte. After the fusion of the sperm cell and the secondary oocyte, changes occur in the cell membrane of the secondary oocyte. There will be rapid depolarization of the cell membrane of the oocyte. The depolarization, it prevents entry of further sperm cells into the oocyte. The depolarization also causes an increase in the intracellular content of the calcium ions. The calcium ions, they stimulate secretion of vesicles containing certain substances to the outside of the secondary oocyte. Those substances remove the receptors for sperm cells, so that after entry of one sperm cell into the secondary oocyte, entry of further sperm cells into the oocyte usually cannot occur. So it prevents polyspermy. After entry of sperm into the secondary oocyte completes its second meiotic division, and it is converted into a functional ovum. Then the nuclei of sperm and ovum, they swell up by absorbing fluid. They become vesicular, and they are known as male and female pronuclei. Ultimately, they will fuse, forming the zygote nucleus. this process of fertilization occurs in the fallopian tube. This shows cleavage of the zygote and formation of morula and blastula. So the fertilization occurs in the fallopian tube. Usually in day 1 after fertilization, the zygote undergoes division. This division process is known as cleavage and it is converted into a two-cell shape. Then in day 2, it is converted into 4-cell stage. The embryo is now 4-cell stage. The cells which are formed from the zygote because of cleavage are known as blastomeres. Then on day 4, a solid mass of blastomeres is formed, almost a spherical structure surrounded by zona pellucida. Now this solid mass of cells is known as morula. As this cleavage is occurring, the embryo gradually moves towards the uterus. Then morula is converted into a blastocyst. On day 5, it is converted into blastocyst. Usually on day 4 or day 5, the embryo enters the uterine cavity. In the uterine cavity, the glands of the uterus, they secrete nutritious fluid. That fluid enters into the space between the blastomeres. On day 5, the zona pellucida, by secreting an enzyme, the embryo hatches out of the zona pellucida. So on day 5 in the diagram, you can see that zona pellucida is not there. The fluid is absorbed into the embryo. It is absorbed into the spaces among the blastomeres. Then by accumulation of fluid, a cavity is formed and the morula is converted into a hollow structure, that is the blastocyst. In the bottom part of the diagram, you can see the blastocyst in section. It is a hollow ball-like structure. It has two types of cells, trophoblast cells and embryoblast cells, or the innermost cells. And the cavity is the blastocyst cavity. It is a fluid-filled cavity. This diagram shows how the ovulation occurring. The egg is being, the released oocyte is being received by the fallopian tube, and you can see it is progressing through the fallopian tube. As it is progressing through the fallopian tube, cleavage occurring. Then to the right, you can see the blastocyst. About day 5, after fertilization, it is entering the uterine cavity. and ultimately it gets attached to the wall of the uterus where it will develop. The attachment of the blastocyst to the uterine wall is known as implantation. Implantation, it occurs usually about 6 days after fertilization. The diagram now you see to the left of your screen is a blastocyst which is attached to the endometrial lining of the uterus. Then the diagram on the right, you can see how the implantation has occurred. When the embryo attaches to the endometrial wall, it attaches in such a manner that the innermost cells, they remain next to the endometrium. In this diagram, the innermost cells, they are shown in blue color. And the other cells, they are the trophoblast. The trophoblast will give rise to the chorion. And it is the innermost cells which will form the embryo proper. Then this is a diagram about 8 days after fertilization. You can see the attached embryo is invading the uterine wall. It is getting embedded in the uterine wall. The trophoblast cells, they differentiate into two cell layers, cytotrophoblast and syncytotrophoblast. Syncytotrophoblast, they are the outer layer of trophoblast without having distinct cell boundaries, whereas the cytotrophoblast, they are inner to the syncytotrophoblast and they have a distinct cell boundary. These trophoblast cells, they secrete enzyme with the help of which the embryo buries into the endometrial lining. These are the different stages, how the embryo is getting embedded in the wall of the uterus. This shows, the diagrams show formation of chorionic villi. The chorionic villi are finger-like projections, so they are derived from the cells of the trophoblast. Both syncytotrophoblast and cytotropic trophoblast, they are involved in formation of this chorionic villi. The chorionic villi subsequently develop blood vessels. The chorionic villi, they extend into the blood spaces. When the embryo is getting embedded in the wall of the uterus, under the influence of the enzymes secreted by the trophoblast cells, the endometrial blood vessels, they are eroded, so that in the embryonic tissue, invading embryonic tissue, blood spaces or lacuna are formed. And as further growth occurs, these lacunae, they fuse together, forming large spaces containing maternal blood. They are known as intervillous spaces. and the villi containing blood vessels, the fetal blood vessels, the embryonic blood vessels, they extend into the intervillar space, so that the villi, they are surrounded by maternal blood, and exchange of material between the blood of the developing embryo or the fetus and the maternal blood can occur. The blood vessels of the chorionic villi, they pass through a connecting stalk called umbilical cord. The umbilical cord actually connects the placenta, developing placenta, with the fetus or embryo. This diagram shows placenta, the constituent of placenta from the embryonic side. You can see chorionic villi, that is the fetal part of the placenta. Adjacent to it, to the left side, is the maternal portion. It is the decidua basalis of the uterus, which forms the maternal portion of placenta. So by contribution of both placental and fetal tissue, by the maternal and fetal tissue, the placenta is formed. The placenta is a pancake-like structure. At the time of birth, that placenta is separated from the uterine wall. Since it is detached from the rest of the uterus at birth, it is known as also as after birth. So this was a brief content of what was there in the unit. Now we'll discuss actually the questions. अच्छा ठीक है, सुनिए ठीक है, এইটা এইপট সাইডটা এটা কভার হয়ে যাଉଛି দুশ্চনি কোশ্চেনটা কোন করিয়া ହଁ ସାର୍ ଏପଟ ସାଇଡ ଟିକେ ବ୍ଲକ ହେଇ ଯାଉଛି ଏଇଟାକୁ କେମିତି ଟିକେ ହଟେଇବା ହଁ
এটা কোনটা ক্যান্সেল হবে বারবারটা কোনটা হাইড করে দিয়েছে ঠিক আছে সেটা সামনে দেখো এখানে থাকতে ঠিক এটা দেখি দিন স্যার সামনে দেখি বাই চান্স সে করে দিয়েছে
না সেইটা রহিব কি
না
okay now it is okay. Thank you okay sir. So now we'll discuss actually the multiple choice questions involving this chapter. First question, as you can see, the walls of the seminiferous tubule contain non-germinal cells. We have different options, out of which the option A is the correct one. That will be the answer. The seminiferous tubule contain both germinal cells and non-germinal cells. The Sertoli cells are the non-germinal cells within the seminiferous tubule. Then the question 2. The column of erectile tissue present along the ventral side of penis. We have options like corpora cavernosa, corpus cavernosum, corpus spongiosum, and none of these. Here the answer will be corpus spongiosum. It is the cylinder of erectile tissue present along the ventral side of the penis. The dorsal ones, two dorsal ones, they are known as corpora cavernosa. Then next question. Which of the following characteristics is not associated with testosterone? We have options as maintenance of secondary sexual characteristics, then responsible for primary sex characteristics, principal androgen, and protein hormone. So here the answer will be D. Testosterone, it is a hormone, but it is not protein. Then fourth one, which of the following is not a function of Sertoli cells? To phagocytize excess formatid cytoplasm as development proceeds. Then to control movement of spermatogenic cells and the release of sperm into the lumen of the seminiferous tubule. Third option is to produce fluid for sperm transport and secrete the hormone inhibin. Fourth option is to regulate the effect of LH. Out of the four, the answer will be D, because Sertoli cells, they do not regulate the effects of LH. Sertoli cells regulate the effects of follicle stimulating hormone, not that of the LH. Then question 5, Leydig cells. Leydig cells are located where? Within seminiferous tubules, in the space between adjacent seminiferous tubules, in the wall of the epididymis, and within the seminal vesicles. The Leydig cells, they are located in the space between adjacent seminiferous tubules. So it is not located within seminiferous tubules. Then question 6. Which of the following male accessory glands is not a paired structure? We have options as seminal gland, prostate gland, vulvourethral gland, and none of these. Of these glands, prostate gland, it is not paired, whereas seminal glands and vulvourethral glands, or the Kapoor glands, they are paired structure. So the answer is B, prostate gland. Then question 7. This is the male copulatory organ, also a passageway for ejaculation of sperm and excretion of urine. We have options as urethra, penis, ejaculatory duct, and ductus deferens. The answer will be penis. Then question 8. This carries the sperm from the scrotum into the abdominopelvic cavity for release by ejaculation. We have options as epididymis, ductus deferens, ejaculatory duct, and urethra? The answer will be ductus deferens. It is the duct which carries the sperm from the epididymis in the scrotum into the abdominopelvic cavity. Then question 9. This surrounds the urethra at the base of the bladder and produces secretions that contribute to the sperm motility and viability. We have answers as prostate gland, Cowper's gland, seminal glands, and none of these. Here the answer will be prostate gland. It is the prostate gland which is present around the base of the urethra. The prostate gland, it secretes an alkaline fluid. The secretion of the prostate gland, it contains citric acid, proteolytic enzymes. It contains pepsinogen, lysozyme, amylase, hyaluronidase, and an antibacterial substance that is seminal plasmin. The prostate gland accounts for 25% of the seminal fluid. The secretion of the prostate gland, it contributes to the sperm motility and viability. Then question 10. Which part ejects sperm into the urethra just before ejaculation? We have options as epididymis, ductus deferens, ejaculatory duct, and none of these. The answer will be ejaculatory duct. It is the ejaculatory duct which ejects sperm into the urethra just before ejaculation. Then next question, question 11. This secretes an alkaline fluid to help neutralize acids in the female reproductive tract. Also, it secretes fructose for use in ATP production by sperm. We have options as prostate gland, Cowper's gland, seminal gland, and none of these. The answer, sir? Right, how it becomes have those?
Dear friends, I request Prakash to take 5 minutes leisure. Now in this time, I want to share with you, have you any doubts regarding any of your academic activities? Apanko school le academic activities ba say related kichhi asubidha hauchi ki? Please tell me. I will try to sort it out. If you have got any type of academic difficulties in your schools, please tell me so that I will try to do it with Director ST or your Assistant Secretary here, Jayashree Madam. So I can talk with Jayashree Madam or I can talk with Director ST if you have got any type of difficulties in academic activities at your school level. Please tell me. Mat kuhନ୍ତୁ. Any of you? Kichi asubidha hochi jodi kuhନ୍ତୁ. I will try to sort it out. Kuhନ୍ତୁ.
No, sir.
Kichi asubidha nahi. Acha, are you?
No, sir.
Acha, are you performing practicals in your schools or not?
Yes, sir.
Yes. Now, very recently, last month, we have made some recording, practical recordings in physics, chemistry, mathematics, botany, zoology, IT, computer science. So that has been recorded. We are trying to edit it and send it to you people. And in the meantime, whether you are aware or not, that we have made 780 hours recording of +2 syllabi in each subject: physics, chemistry, mathematics, computer science, botany, zoology. We are including English. We have in each subject we have made about 30 hours recordings in your syllabi, because you know that the CHSE syllabi and CBSE syllabi are more or less around 99% same. So basing on that, we have already recorded that thing around 6 months back, and we will send it to you, because you can see to our department website, STSC RTI, and not SC and SC development. Sanatana koti achi? SC. Yes. No, you can go to scstrti@gmail.com. In our SCSTRTI website, we have already loaded 30 hours recording for second year students for class 12. SCSTRTI YouTube. You go for SCSTRTI YouTube, and in each subject we have made 30 hours recording of class 12. So for your help, it will fit to your help, and you can ask your students to go and see it, because the experts they have, we have invited various experts and we have recorded, and that is already public. So you can take advantage of this one by going to SCSTRTI YouTube. You can see second year class 12 syllabus. And regarding practicals, we have already done recordings about in each subject 12 practicals, including botany, zoology like spotting or anything, physics, chemistry, everything, and computer science. And that is already under the process of editing. So once that is edited, within a month, we will release it, so that you can take advantage and your students can take advantage of that practical recordings. So you can do that thing also, and that will be practical recordings will be released around 1 month, within 1 month. But theory you can see just now. Any other doubts?
Okay, sir. Thank you.
Any other thing? ଆପଣ ଆଜିଠୁ ଦେଖିପାରିବେ ଯୋଉ ଥିଓରି ପାର୍ଟ ରେକର୍ଡିଂ SCSTRTI ୟୁଟ୍ୟୁବ କୁ ଯାଇକିରି।
Theory ଦେଖିଛୁ sir?
ହଁ କଣ କହିଲେ?
Theory ଆଗରୁ ଦେଖିଛୁ sir, load ହେଇଛି।
ହଁ ଥିଓରି ଲୋଡ ହଲେ ହେଇ ସାରିଲାଣି, ମାସ ଉପରେ ହବ।
Sir sir।
ସେ କହିବାକୁ ଭୁଲି ଯାଇଥିଲି। ଆଉ ଏମିତି ଆଚ୍ଛା ଶୁଣନ୍ତୁ, ଗୋଟେ ପ୍ରୋଫର୍ମା ଆମେ ଛାଡ଼ିଛୁ ଏଥର। ମିନିଷ୍ଟ୍ରି ଅଫ ଟ୍ରାଇବାଲ ଆଫେୟାର୍ସ, ଗଭର୍ଣ୍ଣମେଣ୍ଟ ଅଫ ଇଣ୍ଡିଆ, ସିଏ ଚାହୁଁଛି ଯେ ଯୋଉ ସାର୍ ମାନେ କୋଉ ସ୍କୁଲରେ କୋଉ ସାର୍ ମାନେ ଅଛନ୍ତି, ବିକଜ ତାଙ୍କୁ ଗୋଟେ ଡାଟା ଦବା ପାଇଁ ଆପଣ କ୍ୱାଲିଫିକେସନ ଏବଂ ଆପଣମାନେ ଜାଣିଛନ୍ତି ଆପଣଙ୍କର ଯୋଉ ରେଗୁଲାରାଇଜେସନ ଅଫ ସର୍ଭିସ ବା ପେ ହାଇକ୍, ତାକୁ ରିଲେଟେଡ କରି ସମଥିଙ୍ଗ ୱିଲ୍ ଟେକ୍ ପ୍ଲେସ୍। ସୋ ଯୋଉଗୁଡାକ ଆମେ ପଠେଇଛୁ ଆପଣଙ୍କ ସ୍କୁଲର ସବୁ ଟିଚର ମାନଙ୍କ ନାଁ, କ୍ୱାଲିଫିକେସନ ଏକ୍ସେଟ୍ରା, ଆପଣ ଟିକେ ଜାଣିକି ଆପଣଙ୍କ ଭିତରୁ ଇନିସିଏଟିଭ ନେଇକିରି ଏନିବଡି ମାନେ ସେ ଫିଜିକ୍ସ କେମିଷ୍ଟ୍ରି କରିକିରି ସେଇଟା ଆମ SCSTRTI ର ଆମର ଯୋଉ ୱେବସାଇଟ APMC ରେ ଅଛି ସେଠିକି ପଠାନ୍ତୁ। That will help you, because you will be registered at MOTA level for Government of India for your future point of view. Any other thing? Now yes, କଣ କୁହନ୍ତୁ, କଣ କହୁଛନ୍ତି କୁହନ୍ତୁ ସାର୍।
sir load କରିବୁ sir ସେଟା, ଠିକ ଅଛି।
ଏବଂ ଆପଣ ମାନେ you can take the help of your headmaster। ଆମେ ଛାଡ଼ିଛୁ ସେଟା, headmaster ମାନଙ୍କର ବି ନାଁ ଦେବେ, qualification। Anyway, more time ନଉନି। Now I am again returning Prakash sir to start his deliberation. Prakash sir.
Thank you, sir.
सर आ गुड कथा थिला
कौन कुहतु कुहतु
पळेले
कुहतु सर
सर हमर जो पिला हां सर मो पिला मानकर जो सीबीएसई सर हे जाई ओएमआर बेस्ड प्रैक्टिस करिया पे के ओएमआर सर जदी सुविधा करहंता पिला पे भलो होता
शुणु आमे ओएमआर गत वर्ष करू गले एठी थुआ आइछि वी हैव स्पेंड अराउंड 2 lakhs ऑफ मनी किंतु विधाय माने विधिर विधान तक किछि परीक्षा करियानी एई बर्तन एई जो रूम में चलती सब अलमारी ऊपर गद्दा आइछि ओएमआर सीट परीक्षा करिया तो आमे चेष्टा करबो परीक्षा करिया पे किंतु ओएमआर सीट पब्लिश करे हम दो पे सेकंड में काइला लास्ट ईयर 2 लाख टका पाखापाखि खर्च 1.84 लाख खर्च करे दैट हैज बिकम अ वेस्ट जे तो गवर्नमेंट पूरा बैन कर देला तेणु सते गवर्नमेंट तक कारण से अकाउंट्स रो असुविधा हो तेणु सेटा हमें ओएमआर टेस्ट पे करबो बट द ओएमआर सीट आपन माने आपन जकन हट नै के खाली ऑर्डिनरी जेरॉक्स करे करी पिला को देके परीक्षा कर पारी हमें चेष्टा करे पे कोन है काइला पता सुदा जदि सीबीएसई पाइछि वी विल ट्राई टू डू इट सॉर्ट इट आउट किंतु हमें ओएमआर रो जेतो हमें 2 लाख टका ऑलरेडी इन्वेस्ट ही सरछि तो गवर्नमेंट के राजी होबनी ए मोर आगे ट्राई सब अलमारी वाले को ऊपर पूरा सब anyway we will try I am noting your suggestion we will try if possible Prakash sir
Okay, sir. Thank you. So, coming back to the multiple choice questions. Question 12. Dash is the shared terminal duct of reproductive and urinary systems in male. We have options as epididymis, ductus deferens. Then C, there is a spelling mistake. It should be urethra, and finally the option is ejaculatory duct. The option will be urethra. Then question 13. Semen contains all of the following except mucus, clotting enzymes, substances to lower the pH of uterine environment, and an active clot dissolving enzyme. The answer will be D, an active clot dissolving enzyme. Then 14, secretions of dash contribute maximum to the volume of semen. We have options as bulbourethral glands, seminal vesicles, prostate gland, and epididymis. The answer will be seminal vesicles. There are 2 seminal vesicles, and the secretion of seminal vesicles account for about 60% of the volume of semen. Then question 15. Which sequence best describes the passage of sperm? At 1, we have seminiferous tubule, 2 vas deferens, 3 epididymis, 4 is ejaculatory duct, and 5 is urethra. The option will be B. The sequence will be seminiferous tubules, then epididymis, then vas deferens, then ejaculatory duct, and finally urethra. Then next question, semen has a pH of 6.2 to 6.5, 6.5 to 7, 7.2 to 7.7, then 7.8 to 7.9. The correct option is C, that is from 7.2 to 7.7. That means it is alkaline in nature. Then question 17. Testosterone secretion from Leydig cells is under the control of which hormone? Options are FSH, LH, luteinizing hormone, estrogen, and androgen. The correct answer is luteinizing hormone. Then question 18, it is in the form of assertion and reasoning. The assertion is the testicles of male mammals are suspended in the scrotum. Reasoning, the optimum temperature for spawn production is higher than the normal core body temperature of the organism. We have 4 options. Option A, it says both A and R are correct. That means assertion and reasoning both are correct. Then B, assertion incorrect, but reasoning correct. Option C, assertion correct, but reasoning incorrect. Then D, both assertion and reasoning incorrect. The answer will be C. Because the testicles of male mammals are suspended in the scrotum, it is true, but the reasoning is not correct. Actually, the optimum temperature spawn production is slightly less than the normal body temperature. Then 19, question 19, it is also in the form of assertion and reasoning. The penis contains erectile tissue. This is the assertion. Reasoning, when the male is sexually excited, the erectile tissue is filled with blood, causing the penis to become erect. We have 4 options, out of which option A is the answer, because both assertion and reasoning, they are correct. Then question 20. The fibroblast-like cells called stromal cells, they are located in which part of the ovary? Germinal epithelium of ovary, or tunica albuginea of ovary, or cortex of ovary, or medulla of ovary? The answer will be C, cortex of ovary. Then 21, question 21. The funnel-shaped portion of fallopian tube, that is the oviduct or uterine tube, is known as ampulla, isthmus, infundibulum, or fimbriae. The answer will be C, infundibulum. This funnel-shaped part, infundibulum, it receives the secondary oocyte at the time of ovulation from the ovary. Then question 22, the widest and the longest portion of fallopian tube is, options are isthmus, infundibulum, fimbriae, and ampulla. The answer is ampulla. Ampulla is the widest and the longest portion of fallopian tube. Then question 23. The inferior narrow portion of uterus is known as fundus, cervix, urethral, or body. The answer will be B, cervix. Then question 24. Paraurethral glands or Skene's glands of women are homologous to prostate gland of men, Cowper's gland of men, seminal glands of men, none of these. Answer will be A, prostate of men. Then 25. The greater vestibular glands, also known as Bartholin glands of women, they are homologous to prostate of men, Cowper's gland of men, seminal glands of men, none of these. Answer will be Cowper's gland of men. Then question 26. Labia majora of women are homologous to days of men. Whether they are homologous to scrotum, spongy urethra, membranous urethra, or glans penis. Answer is scrotum A. Labia majora, these are skin folds, part of the external genitalia of human female. They are homologous to the scrotum of men. Then question 27. Graphian follicle occupies a position deep in the ovarian cortex, deep in the ovarian medulla, in the peripheral part of ovarian medulla, and near the surface of ovary. Answer will be D, near the surface of the ovary. As you know, ovarian follicles, they start their development in the cortex. As they grow and their size increases, they move towards the interior of the ovary. Again, as the ovarian follicle matures, it comes to the surface of the ovary for ovulation. That's why the Graphian follicle will be found near the surface of the ovary. Then question 28. The vulva does not include which of the following? We have options as vestibule of vagina, clitoris, mons pubis, and cervix. Out of the four, the answer will be D, that is the cervix. Cervix is not part of the external genitalia or vulva of women. Then question 29. Which of the following is not part of a Graphian follicle? Antrum, liquor folliculi, primary oocyte, secondary oocyte. Out of the four, answer will be C, primary oocyte. As you know, antrum is the cavity in the Graphian follicle, and the fluid which is present in that antrum is antral fluid or liquor folliculi, and the oocyte which is present in the Graphian follicle is secondary oocyte. So primary oocyte will not be there in the Graphian follicle. Then question 30. The middle layer of the uterus is known as perimetrium, myometrium, endometrium, none of these. Answer will be myometrium. The middle layer of the uterus is the myometrium. It is the muscle layer. Then question 31. This refers to the degeneration of oogonia before and after birth. We have options as amenorrhea, atresia, menopause, and none of these. The correct answer is atresia. Then 32. Which of the following structures is the site of spermatogenesis? We have options as prostate, urethra, bulbourethral gland, and seminiferous tubules. The answer is D, seminiferous tubules. Then 33. During spermatogenesis, the diploid daughter cell that begins meiosis is called a primary oocyte, secondary oocyte, spermatid, spermatogonium. The answer will be primary spermatocyte. It is the primary spermatocyte which undergoes first meiotic division to produce secondary spermatocytes. Then 34. Each secondary spermatocyte undergoes the second meiotic division to produce days spermatids. We have options as 2, 4, 6, and 8. Answer will be 2. From each secondary spermatocyte, 2 spermatids are formed. Then 35. Spawns, after being produced, develop motility in epididymis, vas deferens, seminiferous tubule, urethra. The answer will be epididymis. Then question 36. Arrange the following stages into correct sequence. Spermatogonium, spermatid, primary spermatocyte, secondary spermatocyte, and spawn. The correct sequence is given at option D. Spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, and finally spawn. Acrosome of spawn contains DNA, mitochondria, axial filament, and hydrolytic enzymes. The answer will be D, hydrolytic enzymes. Then question 38. Gametogenesis requires the conclusion of meiosis II. After end of meiosis II, the functional gametes are produced. When does this occur in females? During fetal development, at the onset of puberty, after fertilization, after implantation. The answer will be C, after fertilization. Then 39. In human females, egg is released from ovary as primary oocyte, secondary oocyte, ovum, or oogonium. The answer will be B, secondary oocyte. Then 40. Which statement about human oocytes is true? By birth, the human female infant has produced a lifetime supply of oocytes. At the onset of puberty, ovarian follicles produce new oocytes in response to hormonal stimulation. At the onset of menopause, a woman stops producing oocytes. Oocytes are produced by women throughout adolescence. The option will be A. By birth, the human female infant has produced a lifetime supply of oocytes. Then 41. During oogenesis in mammals, the second meiotic division occurs. Options are in the formation of primary oocyte, in the formation of secondary oocyte before ovulation, after fertilization. Answer will be D, after fertilization. 42. Which of the following cells is haploid? Primary oocyte, secondary oocyte, oogonium, and follicle cells. Answer is C, secondary oocyte. Then question 43. During ovulation, entire Graafian follicle is released. Only secondary oocyte is released. Secondary oocyte along with zona pellucida is released. Secondary oocyte along with zona pellucida and corona radiata is released. Correct answer is option D. That is, secondary oocyte is released along with zona pellucida and corona radiata. Then question 44. During oogenesis, the role of polar bodies is to receive the haploid set of chromosomes, to slow down the process of oogenesis, to nourish the developing oocytes, none of these. Answer is option A, that is to receive a haploid set of chromosomes. Other than this, polar bodies have no more function. Question 45. After ovulation, the remaining portion of Graafian follicle develops into a temporary endocrine gland called corpus hemorrhagicum, corpus luteum, corpus albicans, and antrum. Answer will be B, corpus luteum. Then 46, spermatogenesis and oogenesis differ in that option A is spermatogenesis produces gametes with greater store of raw materials than those produced by oogenesis. Spermatocytes remain in prophase of first meiotic division longer than oocytes. Oogenesis produces 4 equally functional haploid cells for meiotic event and spermatogenesis does not. Spermatogenesis produces many gametes with meager energy reserves, whereas oogenesis produces relatively few well-provisioned gametes. 46, the option D is the answer. Then 47, the corpus luteum degenerates if fertilization occurs, develops in the pre-ovulatory phase, is maintained by prostaglandins, serves as a temporary endocrine gland. Correct option is D, serves as a temporary endocrine gland. It secretes estrogen and progesterone. Then 48 question. After ovulation, the secondary oocyte enters the ovary or corpus luteum or cervix or oviduct. Correct answer is D, oviduct. Then 49. Days of Graafian follicles secrete female sex hormones. We have option says the inner part of theca and granulosa cells, only granulosa cells, the outer and inner parts of theca, the outer parts of theca and granulosa cells. The answer is D. Then question 50. Ovulation usually occurs on dash day in a 28-day cycle. We have options as 12, 13, 14 and 15. Answer is C, 14. 51 question. Dash surge brings about ovulation. Luteinizing hormone, follicle stimulating hormone, estrogen and progesterone. The answer is A, that is LH. Then 52, the layer of uterine lining that is partially shed during each monthly cycle is perimetrium, myometrium, endometrium, none of these. Correct answer is C, endometrium. Question 53, the endometrium, 4 options we have, is the muscle layer of uterus. B is the thickest during pre-ovulatory phase, is the site of embryo implantation. D is lines the vagina. Correct answer is C, is the site of embryo implantation. Then 54, the hormone that reaches its highest level during post-ovulatory phase is, we have 4 options, progesterone, estrogen, FSH and LH. The answer is A, progesterone. Then 55, which of following hormones is not secreted by corpus luteum? Progesterone, estrogen, relaxin, luteinizing hormone. The correct answer is D, luteinizing hormone. It is not secreted by corpus luteum. Then 56, if the released oocyte is not fertilized, the corpus luteum has a lifespan of only 4 options we have, 1 week, 2 weeks, 3 weeks, 4 weeks. Correct answer is option B, that is 2 weeks. Then 57, absence of menstruation is known as amenorrhea, atresia, menopause, none of these. Correct answer will be option A, amenorrhea. Then 58, peak secretion of estrogen hormone occurs. 4 options we have. A is during all phases of menstrual cycle, at the time of ovulation, just before ovulation, in mid-luteal phase. Correct option is C, that is just before evolution, just before ovulation. Then question 59, in humans, fertilization occurs in the dash and implantation of the zygote occurs in the dash. Option A, seminiferous tubules, uterus. Option B, vagina, oviduct. C, oviduct, uterus. D, urethra, uterus. Correct option is C, that is oviduct. Fertilization occurs in the oviduct and implantation occurs in the uterus. Then question 60. Fertilization involves all of the following except option A, equal contributions of cell organelles from sperm and egg. B. Joining of sperm and egg haploid nuclei. C. Sperm binding to specific sites on the egg surface. D. Metabolic activation of the egg. The correct answer is A, option A, that is equal contribution of cell organelles from sperm and egg. Then question 61. Mutations that affect proteins in the acrosome would impede which of the following functions? Options are fertilization, meiosis, implantation, semen production. Answer will be A, fertilization. Then 62. The viability of spermatozoa in the female genital tract is up to days hours. 6 to 12 hours, or 12 to 24, or 24 to 48, or 48 to 72. The option is 24 to 48 hours. That means spermatozoa can survive in the female genital tract up to 48 hours. Then question 63. A secondary oocyte is viable for about days hours after ovulation. We have options as 12 hours, 24 hours, 36 hours, and 42 hours, 48 hours. The correct option is B, that is 24 hours. Then 64. Theca cells of ovarian follicle are derived from germinal epithelium, granulosa cells, zona pellucida, and stromal cells. Answer is D, stromal cells. Stromal cells of the ovarian cortex. Then 65. The transport of spermatozoon from cervix to fallopian tube takes about 5 to 10 minutes, 10 to 30 minutes, 30 to 60 minutes, 60 to 90 minutes. The correct answer is 30 to 60 minutes. Then question 66. The first sign of pregnancy is enlargement of abdomen, rise in basal body temperature, lack of human chorionic gonadotropin in urine, absence of menstrual bleeding. The correct option will be D, that is absence of menstrual bleeding. Then 67. The main function of the acrosome reaction is to activate the egg, improve sperm motility, prevent interspecific fertilization, facilitate penetration of the egg coverings of the sperm. Correct option is D, that is to facilitate penetration of the egg coverings by the sperm. Then 68. Capacitation of sperm occurs in epididymis, vas deferens, urethra, genital tract of female. Correct option is D, that is the genital tract of female. Then 69. Penetration of the zona pellucida is facilitated by enzymes in the sperm's acrosome, nucleus, middle piece, tail. Correct option is A, that is acrosome. Then question 70. The human zygote divides to become a two-celled embryo. Days of fertilization, within days hours of fertilization. We have option says within 12 hours, after 24 hours, after 36 hours, after 48 hours. The correct option is B, that is after 24 hours. Then question 71, the cleavage of fertilized egg starts in the oviduct, ovary, uterus, vagina. The correct option is A, oviduct. Fertilization occurs in the oviduct. Also, the process of cleavage starts in the oviduct. Then 72, the process in which embryo embeds in the endometrium of the uterus is known as implantation, pregnancy, placentation, none of these. Correct option is A, that is implantation. Then 73, which cells of a blastocyst secrete human chorionic gonadotropin? All the cells of inner mass, trophoblast cells, none of these. The correct option will be C, that is the trophoblast cells. It is the trophoblast cells which secrete human chorionic gonadotropic hormone. Then question 74. After implantation, the endometrium becomes modified and is known as decidua, placenta, umbilical cord, chorionic villi. The answer will be A, that is decidua. Then 75, which of the following consists of both fetal and maternal tissue? We have options as umbilical cord, placenta, allantois, and yolk sac. The correct option is B, that is placenta. Then question 76. Dash contains the vascular connections between mother and fetus, placenta, amnion, chorionic villi, umbilical cord. The correct option is D, that is umbilical cord. It is the actual vascular connection between mother and fetus. Then 77. One of the several hormones produced by placenta is human chorionic gonadotropin. It maintains the activity of corpus luteum, promotes degeneration of corpus luteum, promotes lactation, inhibits FSH. The correct option is A, it maintains the activity of corpus luteum. Then question 78, placenta does not secrete human chorionic gonadotropin, human placental lactogen, progesterone, and finally prolactin. The correct option is D, that is prolactin. Placenta does not secrete prolactin hormone. Then question 79. Dash stimulates the corpus luteum to continue production of progesterone and estrogen. Is it human chorionic somatomamotropin, human chorionic gonadotropin, prolactin, or relaxin? Correct option is A, human chorionic somatomamotropin. Then 80, the human gestation period That is the duration of pregnancy average, averages days from the time of fertilization to the birth of the baby. So the average gestation period is how many days? 266 days, 290 days, 256 days, or 296 days. The correct option is A, that is 266 days. Then 81, days increases the flexibility of the pubic symphysis and helps dilate the uterine cervix during labor. Options are oxytocin, human chorionic gonadotropin, prolactin, and relaxin. For question 81, the correct answer is relaxin, that is option D. Then question 82, days helps prepare mammary glands for lactation. Options are oxytocin, human chorionic somatomammatropin, human chorionic gonadotropin. D is prolactin. Correct option is D, prolactin. Then 83, days stimulates uterine contractions and also responsible for milk ejection reflex. Oxytocin, human chorionic gonadotropin, prolactin, relaxin. The correct option is oxytocin. Then question 84, days promotes milk synthesis and secretion, also inhibited by progesterone during pregnancy. Options are oxytocin, human chorionic somatomammatropin, human chorionic gonadotropin, and prolactin. For 84, the correct option is D, that is prolactin. Then 85, the pregnancy test is based on the detection of four options are LH, HCG, progesterone, and estrogen. The correct option is B, that is HCG, human chorionic gonadotropin. Then 86, the ovarian follicle that ruptures at the time of ovulation is promptly filled with blood, forming corpus hemorrhagicum, corpus luteum, corpus albicans, none of these. The correct option is A, that is corpus hemorrhagicum. 87, the discharge of secondary oocyte from Graafian follicle is known as, options are abortion, oogenesis, ovulation, fertilization. The correct option is C, that is ovulation. Then 88, it is in the form of assertion and reasoning. Assertion, spermatozoa after being released are not immediately ready for fertilization. Reasoning, without undergoing capacitation, spermatozoa cannot fertilize oocyte. Here the option A is the correct answer because both assertion and reasoning are correct. Then 89, assertion, during a menstruation phase, blood, tissue fluid, mucus, and epithelial cells are shed from endometrium. Reasoning is, menstruation is caused by sudden decline in estrogen and progesterone. The correct option is A, that is both assertion and reasoning are correct. And this is the final question. Assertion is pre-ovulatory phase of menstrual cycle is known as proliferative phase. Reasoning, during pre-ovulatory phase, the endometrium and myometrium increase in thickness. Here, the correct answer is option C. So here the assertion is correct, but the reasoning is incorrect. So these are some of the questions I wanted to discuss with you regarding human reproduction. Thank you.
And dear friends, we come to the end of this session. Thank you, Dr. Paricha. And these questions which he discussed will be shared with you within a few days so that you can give it to your students also at your part, because you know that the first term and examination will mostly MCQ type of questions. Again, I am very much thankful to Dr. Paricha, and just I am giving you 5 or 10 minutes break so that we can start the next session after 5 or 10 minutes. So till then, for 5 to 10 minutes, let's have the break and we can have the next session. Sir. Yes, yes, yes. Kuantu, madam.
Sir, ଆମେ ଯୋଉ performance sir ଦିଏ ଯିବ sir TGT ମାନଙ୍କର sir ଦିଏ ଯିବ ନା PGT sir ସମସ୍ତଙ୍କ ଦିଏ ଯିବ sir ସେ ଯୋଉ performance ରେ
କୋଟା ମୁଁ ବୁଝି ପାରିନି କଣ क्वेश्चन unable to hear you please ଆପଣଙ୍କର ସେ ପାଖକୁ କିଛି ହଁ କୁହନ୍ତୁ madam
sir କହୁଥିଲେ ଯେ sir ଆମର PGT ସମସ୍ତ ଦିଏ ଯିବ ତ ସେ ଯୋଉ performance
मैडम आपण मानक डिटेल्स आगुर आसी जाईची एबे दरकार नै हमर डाटाबेस मागिथलु एमआरएस रु आसी जाईची
तो एमआरएस दरकार नै एचएसएस हम करबो ना आपण कथा ब्रेक करूची मैडम मते परे नले फोन करे कहबे मो पर्सनल मोबाइल नंबर रे मते परे कहबे कारण आपण कथा सुबुजने ब्रेक करूची ओके Great. Dear colleagues, recording here, recording. Dear colleagues, we now move to the second session, and on my left is Dr. Bandita Ratha. She has done her PhD in biotechnology also from Life Science Institute, and she is now associate here, assistant professor in Rajdhani College, Bhubaneswar. So I thought she is the fittest person to be given this topic. And as you, the teachers, you may be not knowing much regarding the biotechnology part, so you should take full advantage of her talk or his deliberation. And she has prepared a very nice PDF as well as questionnaire answering subjective type question, MCQ type of question. And we will also share you with all these type of things within very few days, day or after tomorrow, something like that. So I don't want to become a bar. Now I request Dr. Bandita Ratha to start her deliberation. Dr. Ratha, please.
Thank you so much, sir.
She's there.
Good morning to all my colleagues present in this orientation program. I feel most of you must have been there during my last presentation where we have dealt with biotechnology. So most of my questions that I have done over here are based on those PPTs, are based on various books for +2 which is available, and also certain questions that are entrance exam based. So I feel I've covered almost all the aspects of this first topic, which is your biotechnology principle and processes. So let me first share you a PPT. Thank you. So I have dealt with this one. Those of you are present during the last orientation, I have already dealt with this one. So first is we will be going through all the questions. The first half will be going to the MCQ. So it will be dealing with the principle and processes of biotechnology, which is genetic engineering or your recombinant DNA technology. So in this, we have gone through the initial diagram or to the structure of our DNA. How is our DNA? What are the various bases which are present regarding the basic, because biotechnology is nothing but the basic of our DNA and RNA. If we know our DNA and RNA basic, how phosphodiester bond, what is phosphodiester bond, how is this phosphodiester bond formed, if we know that one, then this biotechnology is basically nothing. It is based on this bonds of DNA and RNA. So let me go to the question, to the MCQ questions. Okay. So first, like I have prepared around 80 or so odd question over here covering the entire biotechnology and processes. General questions I have given, some questions which are repeated in different modes so that student won't get confused. Those also I have tried to incorporate. So first going to combining the DNAs of two species into a single DNA molecule is known as. I'll be starting from the basics. So we all know that when we are combining DNA from two different sources, like from human being or a bacteria, we are taking a gene from a human being and we are inserting it into the plasmid of a bacteria and introducing it into the bacteria. So this is, we are recombining the DNA. So this is what is known as the recombinant DNA technology. So various examples have been given over here. So one might be a bit baffling. Sorry, I'm so sorry. one might be a bit baffling for the students, that is crossing over. Okay, genetic recombination, crossing over, gene amplification. Genetic recombination is there, but genetic recombination is occurring in inside in our normal processes within our body also. Genetic recombination is occurring inside our body. Crossover is occurring inside our body. But over here, the genetic recombination, it does not basically mean that this recombination is taking place between two different species. It is something which is taking place within us, within the same species and within the same individual. So that is a genetic recombination. Crossing over is also taking place within the same individual. And gene amplification, when a single gene is being amplified or it increases in number. There is a technology known as PCR. So when we are increasing the number of DNA, that is known as a gene amplification. So the correct answer is recombinant DNA technology. So students should be aware of this genetic recombination, crossing over, which might be a bit confusing for them. Then construction of recombinant DNA involves cleaving and joining the DNA fragments with endonuclease, cleaving DNA segment with endonuclease and rejoining it with ligase, cleaving and rejoining DNA segment with ligase, cleaving DNA segment with ligase and rejoining it with endonuclease. So when we will go to the processes, of let me just have it. So if we are going into the processes of recombinant DNA technology or steps of recombinant DNA technology, so first what is happening is generation of your DNA fragment. First, we are cutting the DNA, okay? Then we are insertion of selected DNA into a cloning vector to create a recombinant DNA or a chimeric DNA. So the first thing that is occurring is we are cutting. So we can see over here, they have given a peach color rounded structure over here. So this not jet structure basically, this haphazard line that we are seeing over here, the DNA is cut over here. So first we need to cut the DNA, then this is a DNA from other source. So this is a DNA from another source. And this is DNA from this is basically a plasmid. So plasmid, we are getting it from bacteria. So we are taking the DNA from any other source, may it be a human being, may it be a mouse, may it be a Drosophila, anyone. We are taking this gene of interest or this particular DNA segment, and then we are inserting it into the plasmid that belongs to a bacteria. Okay, so first we need to cut the DNA, and second, we need to join the foreign DNA into the plasmid or the vector. Okay, so we require two enzymes. So for cutting, we require restriction endonuclease. Always for cutting purpose, we require restriction endonuclease. After cutting, then we join or we ligate. Ligate means joining. The literary meaning of ligate is joining. So after your cutting with restriction endonuclease, we join it with the help of an enzyme which is known as a DNA ligase. Okay, so the correct answer over here will be so the correct answer here is cleave DNA segment. Sorry, this is a wrong one. I'll correct it. It is a cleaving the DNA segment with the endonuclease and rejoining it with ligase. Cleaving the DNA segment with the, it's done by mistake. It is the correct answer is cleaving the DNA. Just a moment. Let me have a Word one. It will be even more helpful. Let me open the Word file. So the correct answer is you're cleaving the DNA segment with your endonuclease and rejoining it with the ligase. Then who developed the chemical techniques to synthesize polynucleotide? Chemically, that is, we are doing it outside. This synthesis of the DNA, we are doing it outside with the help of a machine. The DNA is not synthesized within our body. It is being done outside with the help of a machine. Then who was first to do that? It was Dr. Hargobind Khurana. It was Dr. Hargobind Khurana, an Indian American. He is basically having had a American citizenship. So it was Dr. Hargobind Khurana who first chemically synthesized. Baki synthesis of DNA, mane ama body bhitere DNA synthesize hochi. Kintu DNA ama body bahare je chemically je DNA taku kiye first synthesize kartile se hochante Dr. Hargobind Khurana. Okay. So, say kebe kartile. So the second question is a follow-up of the first question. In 1979, Dr. Khurana synthesized the first functional synthetic gene coding for dash of E. coli. Hargobind Khurana 1979 re first functional एठी भी गोटे confusion अछि से दीटा gene करथिले से दीटा gene तैयारी करथिले गोटे जोड़ा तैयारी करथिले से फंक्शनल नथिला माने से काम कर नथिला से जीन टाको जेते बेले ई कॉली रे इंट्रोड्यूस करा हितला से जीन रो प्रोटीन तैयारी हेई पार नथिला से जीन रो आरएनए हेलानी आरएनए रो प्रोटीन ता तैयारी हेलानी तो सेति पय सांकर फर्स्ट जीन ता नॉन फंक्शनल थिला तापरे से ता ऊपर वर्क करके अगेन व्हाट ही डिड वाज ही सिंथेसाइज द फर्स्ट फंक्शनल जीन. So, ago gene ta jo se karthile, it was non-functional. The second gene was functional. So, dita jako bhitru first functional gene was the second one. So, say second gene ro na ta kona, it was tyrosine tRNA. Seta E. coli ro tyrosine tRNA. First gene ta se jo synthesize karthile, kintu seta kama kar nathila, that is your East alanine tRNA. The first East alanine tRNA karithile synthesis करथिले माने निजे बाहर केमिकली सिंथेसाइज करथिले किंतु सेडा काम करनथला फंक्शनल वन इज अ टायरोसिन tRNA. Okay, so now coming to who the first recombinant DNA was synthesized by. Those who were present, we had already been into this one. During the last orientation, we have already dealt with this one. It was Stanley Cohen and Herbert Bohr who made the first recombinant DNA. So what basically they did was they extracted the tetracycline R gene. Tetracycline R gene means tetracycline resistance gene. जोटा के साल्मोनेला टाइफिमुरियम रे थाए से टेट्रासाइक्लिन रेजिस्टेंट जीन टाकू से माने एक्सट्रैक्ट करथिले तापरे ई कॉली इस्चुरिकिया कॉली रु से माने प्लाज्मिड ता बाहर करथिले. प्लाज्मिड इज अ राउंडेड स्ट्रक्चर. Plasmid is always a double stranded DNA and it is circular. Okay, ए प्लाज्मिड टाकू बाहर करके से माने रेस्ट्रिक्शन एंडोन्यूक्लियस रे कट करथिले. रेस्ट्रिक्शन एंडोन्यूक्लियस रे कट कला परे हमर जो साल्मोनेला टाइफिमुरियम रु से माने टेट्रासाइक्लिन रेजिस्टेंट जीन को इस्चुरिया कॉली रो प्लाज्मिड भितरे इंट्रोड्यूस करथिले बा लाइगेट करथिले. एटा जो फॉर्मूला दिस इज द फर्स्ट रिकॉम्बिनेंट मॉलिक्यूल बाय स्टैनली कोहेन and Herbert Bohr, and this recombinant molecule was introduced into E. coli. And after introduction into E. coli, a bacterium, what it did was it reproduced the tetracycline resistant protein. से E. coli भितरे तारो mRNA तैयारी कला से E. coli रो प्लाज्मिड भितरे इंसर्ट हे जाईची से E. coli भितरु को जाई साला परे ट्रांसफॉर्मेशन हे साला परे से सेटू तारो फर्स्ट mRNA सिंथेसाइज हला. mRNA सिंथेसाइज हला परे देन प्रोटीन सिंथेसिस हे की फाइनल प्रोटीन प्रोडक्ट व्हिच इज tetracycline resistant protein produce हला. Usually Escherichia coli tetracycline resistant नहीं. आमे जदि native normal Escherichia coli भितरे आमे जदि tetracycline आमे जदि पकेबा से माने मरि जिवे. किंतु एटा जो recombinant DNA technology द्वारा हला this was tetracycline resistant. So the answer is Herbert Boyer and Stanley Cohen. So first recombinant DNA molecule, again say question ro tike epart separt kariki puni ethi diya hai. The first recombinant DNA molecule was synthesized in the year 1972. ये जो टेट्रासाइक्लिन रेजिस्टेंट प्लाज्मिड टा जो फॉर्म हेतला सेटा 1972 रे आ मु जोठी भाबी ची टिके टिके एक्सप्लेनेशन दो पीपीटी इज ऑलरेडी अवेलेबल मु जोठी भाबी ची टिके एक्स्ट्रा मो एक्सप्लेनेशन मध्य ऐड कर दी ची. So which observation was made by Everett, MacLeod and McCarthy? से माने ए तीन जन साइंटिस्ट कोन ऑब्जर्व करथिले. से फर्स्ट ऑब्जर्व करथिले जे डीएनए कैन बी टेकन अप फ्रॉम अ मीडियम. माने डीएनए कैन बी टेकन अप फ्रॉम अ मीडियम माने आमे जदी यूजुअली डीएनए टा गोटे ऑर्गेनिज्म भितरे थाए. आमे जदी से ऑर्गेनिज्म रो डीएनए टा बाहर करी की आउ गोटे ऑर्गेनिज्म सपोज इमेजिन सेल कल्चर चलीची. सेल कल्चर भितरे आमे जदी लिवर सेल्स उडा को नेचे आर्टिफिशियल कल्चर्स आछि गोटे पेट्री प्लेट रे लिवर सेल उडा अछि. मु जदी बाहरु किछिटा प्लाज्मिड ऐड करबी आ प्रॉपर कंडीशन देबी तो कोन हबो से लिवर सेल उडा को से बाहर से प्लाज्मिड टा लिवर सेल रो नुहे किंतु से माने कोन करिवे जो बाहरु जो प्लाज्मिड टा जो मु जो ऐड करिछि सेई मीडियम भितरु को सेई सेल उडा को से मीडियम माने सराउंडिंग से सेल ग्रो करबा पई आमे से मानकु गोटे मीडिया देइताउ. से मीडिया भितरे जहां नेसेसरी जिंस गोटे सेल ग्रो करबा पई आम बॉडी भितरे दरकार से मीडिया भितरे भी एग्जैक्टली सेई सबु जिंस गुडा को थाए. ओके से मीडिया सहित मु जदी से प्लाज्मिड टा देली आ प्रॉपर कंडीशन मु प्रोवाइड कली से लिवर सेल गुडा को कोन करिवे से लिवर सेल गुडा को नै जाय पारिबे से प्लाज्मिड टा. So DNA आम मीडियम रो DNA easily गोटे सेल नै पारिबो. DNA can be taken up from the medium by a cell एठी भी रही पारे. So medium रो DNA easily नै पारिबो. के फर्स्ट ऑब्जर्व करथले it was Avery, MacLeod and McCarthy. Okay, so manipulation of DNA to change structure. तो एठी कंफ्यूजन करबा पई एठी जेनेटिक इंजीनियरिंग माइक्रोबायोलॉजी फिजियोलॉजी न्यूरोलॉजी अछि किंतु बायोटेक्नोलॉजी भी आस जाय पारे कंफ्यूज करबा पई. किंतु आमे एनी मैनिपुलेशन एनी केमिकल चेंजेस दैट वी आर डूइंग टू अ डीएनए एनी केमिकल चेंजेस नॉट नेसेसरली आमे रिकॉम्बिनेंट डीएनए रे आमे कोन करछि आमे बाहर माने आउ गोटे स्पीसीज रो डीएनए आनी की आ गोटे स्पीसीज भितरे पुरोचे दीटा डिफरेंट सोर्स रो हमें डीएनए कंबाइन कोचे सेति पे हमें सेटा को रिकॉम्बिनेंट डीएनए टेक्नोलॉजी कोचे. किंतु जेनेटिक इंजीनियरिंग आमे जेते बेले फोर्सफुली जानी सुनी की गोटे डीएनए टा को केमिकली अल्टर कोचे तार नेटिव डीएनए अछि नेचुरल डीएनए अछि किंतु आमे सेटा को केमिकली जेते बेले अल्टर कोचे आमे सेटा को कहबा जेनेटिक इंजीनियरिंग आमे सेटा को रिकॉम्बिनेंट डीएनए कहबा. Recombinant DNA केते बेले होबा जेते बेले we are taking DNA from two different sources and combining them. Okay, so this answer is the genetic engineering. Then recombinant DNA is a DNA molecule carrying new combinations of protein, gene, RNA. None of this. DNA जदि अछि हमर gene ही रहिबो because DNA is तारो केमिकल डीएनए जदि जीन रो केमिकल स्ट्रक्चर टा होची डीएनए डीऑक्सी राइबो न्यूक्लिक एसिड. ओके से डीएनए रो हमरा प्रोटीन होची डीएनए रो फर्स्ट आरएनए होची तापरे प्रोटीन होची. सो इट इज ऑलवेज जीन. इट इज न्यू कॉम्बिनेशंस ऑफ जीन. ओके नाउ गोइंग टू द 10th one. Natural genetic engineer is Pseudomonas natural, Pseudomonas putidus, Escherichia coli, Bacillus subtilis. ए सब गुडा को आमे अल्टर करोचे तापरे माने आमे एमानकर डीएनए को अल्टर करेला परे माने रिकॉम्बिनेंट होउचंती किंतु आमर नेचुरल होची एग्रोबैक्टीरियम ट्यूमिफेशिया. ता भितरे एमितिया गोटे प्लाज्मिड अछि से प्लाज्मिड टीआईएफ प्लाज्मिड आमे कहूचे जोटा कि आमे नेक्स्ट चैप्टर आम नेक्स्ट क्लास रे आमे डील करिबा. से टीआई प्लाज्मिड भितरे से आपे आपे से कौन करे से गोटे जीन नहीं जाए आउट ट्यूमर फॉर्मेशन रे से हेल्प करे okay so এটা naturally हुए naturally plant tumor because of agrobacterium tumefacium plant tumor এটা naturally हेई था सेति पय हमर natural genetic engineer होचंती agrobacterium tumefacium आ अन्य species उडा को आमे alter करचे according to our own needs. Then coming to the 11th question, the enzymes only used in genetic engineering. Enzymes only used in genetic engineering. Restriction endonuclease अच्छी, ligase अच्छी, ligase polymerase अच्छी, restriction endonuclease polymerase अच्छी, endonuclease ligase अच्छी. So एई जाकरे माने almost all these things might be used for recombinant DNA technology. किंतु genetic engineering पाई आमे जत्ते बड़े देखुचे even for recombinant DNA technology आमे खाली endonuclease केबे भी use करूने आमे खाली endonuclease केबे भी use करूने genetic engineering किबा recombinant DNA technology पाई जदि हमको दरकार restriction endonuclease ता सदा बड़े दरकार endonuclease गुडाय प्रकार अछि. Let me show you this slide. So basically we had dealt with an outline of the steps of recombinant DNA technology. First, आमे generate हमको जो desired piece of DNA दरकार आमे से desired piece of DNA को isolate करचे एटा होचे vector भितरे then we are inserting the DNA, we are introducing the recombinant DNA into a host and we are cloning, then we are selecting माने आमरो आमे गोटे दीटा एमिति वेक्टर नहीं ना थाओ आमे मिलियंस ऑफ वेक्टर नहीं थाओ गोटे एक्सपेरिमेंट तो सेतिरे से मिलियंस ऑफ वेक्टर भितरु किछिटा वेक्टर ही ए रिकॉम्बिनेंट डीएनए नहीं थी तो आमरो गोटे सिलेक्शन प्रोसेस एई थाए से सिलेक्शन प्रोसेस रे आमे देखु जे को वेक्टर भितरे ए रिकॉम्बिनेंट डीएनए टा अछि तो सेई वेक्टर सेई होस्ट को आमे सिलेक्ट करके बाहर करनो आ तारो क्लोनिंग माने ताको मल्टीप्लाई हेबा पे दो तो गुडाए आम पाखरे रही गला होस्ट जा भितरे की रिकॉम्बिनेंट डीएनए टा अछि आ सेटा आमे प्रिजर्व कर नै परिबा फॉर अ फर्दर एक्सपेरिमेंट्स. ओके थैंक यू. ओके लेट अस गो टू द नेक्स्ट स्लाइड. So these are basically the tools of recombinant DNA technology. Amara guda enzyme achanti in enzyme, lysing enzyme, cleaving enzyme, synthesizing enzyme, DNA ligase, alkaline phosphatase. So a gote enzyme को भी नहीं कि जोटा कि मु include करची question उड़ा का multiple choice रे include करची. So enzyme needed for opening the cell or dissolving the cell wall of plant. आमे cell टा को open करिबा पे गोटे cell अछि आम DNA टा सेल भीतर अछि सो सेल टा आमे से डीएनए को बाहर करिबा पे आइसोलेट करिबा पे फर्स्ट वी नीड टू डिस्टर्ब द प्लाज्मा मेम्ब्रेन और सेल वॉल जदि if it is a bacterium cell wall, if it is a plant cell wall हमको डिस्टर्ब करिबा दरकार तापरे सेल मेम्ब्रेन और प्लाज्मा मेम्ब्रेन टा को हमको डिस्टर्ब करिबा दरकार सो डिस्टर्ब करिबा पय हमर सदा बेले यूज है लाइसिंग एंजाइम तापरे स्पेसिफिकली हमें जदि आसी जिबा it might also be asked that what lysing enzyme is used to dissolve the bacterial cell wall. Then it is lysozyme. So lysozyme is also present in our tears. आमा आखिरु लुह जो बाहरे सेतिरे भी lysozyme अछि. How it is helping? आमर आखिटा पूरा open अछि. Except for while we are sleeping or when we are blinking for microsecond. Okay. So say समय छाला परे आमा आखि पूरा open अछि. So आमर air रे जिते bacteria घूरी बुलु चंती सेटा आमा आखि को जाई पारे. So से bacteria ओडा को lyse करिबा पे ताको मारी दबा पे आमर आखि रो लुह जो से create हुए सेठी आमर lysozyme गोडा को अछन. Okay, so cleaving enzyme आमे सेते बेले डील करथुले एंडोन्यूक्लियस और रेस्ट्रिक्शन एंडोन्यूक्लियस रो डिस्टिंक्शन आमे डील करथुले. सो हमर क्लीविंग एंजाइम डीएनए को कट करिबा पे हम पाखरे बहुत प्रकार रो एंजाइम अछंती. एक्सोन्यूक्लियस, endonuclease, restriction endonuclease. I think I have also included the question regarding these also. So this exonuclease कौन करिबो amara DNA ta rahila, this is a DNA, so it will be cleaving the DNA from outside 5 prime end ru se 5 prime end ru DNA ta ku kati kati jibo किंतु केबे भी ए मझी रीजन रो एक्सोन्यूक्लियस केबे एक्सो माने बाहर बाहर माने से सदा बेले बाहर ए एंजाइम ता सदा बेले बाहर न्यूक्लियस मींस एनी एंजाइम दैट इज कटिंग द डीएनए और खाली डीएनए नहीं enzymes which cut the DNA and RNA, they are known as a nucleus. Okay, enzyme which is ekire asipare enzymes that are cutting the DNA and RNA is known as DAS. They are the nucleases. Okay, so a nucleases jetebele exonucleus kono jetebele se baharu DNA ta ku kate. Ethi dekhantu baharu DNA ta ku katichi. तो सेइथिपाई एटा हला एक्सोन्यूक्लियस एंडोन्यूक्लियस कोन एंडोन्यूक्लियस को भी जगह रे से भितरु काटिबो एठी देखंतु से गोटे इंटीरियर रीजन रु डीएनए रो गोटे फ्रैगमेंट पे काटिनला देन व्हाट इज दिस restriction endonuclease this restriction endonuclease is an enzyme that will cleave the bond within it will cleave the bond within भितरे ही एंडोन्यूक्लियस माने से डीएनए भितरे ही काटला किंतु गोटे स्पेसिफिक सीक्वेंस रे ही से काटिबो से गोटे स्पेसिफिक सीक्वेंस रे एने तेने कोठी भी काटी दबनी से गोटे एटा सॉरी एटा खाली एंडोन्यूक्लियस देची एंडोन्यूक्लियस इन ग्रॉस से भितरु काटिला भितर डीएनए रु से काटिला सो रेस्ट्रिक्शन एंडोन्यूक्लियस ता कोन एटा होची रेस्ट्रिक्शन एंडोन्यूक्लियस आल्सो नोन एज अ मॉलिक्यूलर सीजर्स. They are also known as a molecular scissors. I have included in this bits also. They are known as molecular scissors, molecular scalpels भी हुआ हुआ है समानों molecular scissors or molecular scalpels. So restriction enzyme they are isolated from bacteria. सेमाने आमे सेमानों को बैक्टीरिया रु आमे पाइचे restriction enzyme bacterial enzyme सेमाने खाली बैक्टीरिया रे ही अछंती आउ सेमाने खाली restriction site रे ही कट करन specific site तांकर थाए they will cut at restriction site जेमिति कि HIN3 गोटे restriction enzyme. This was also the first restriction enzyme to be isolated. This was also the first restriction enzyme to be isolated. एटा भी मु question answer रे मु एटा को भी deal करछि first restriction enzyme to be isolated HIN3 ECOR1 ZABA1 PSD1 गुडाए मु देछि so सेथरु हमर HIN3 is the first restriction enzyme. से कोटि काटे से गोटे palindromic sequence रे काटे palindromic sequence ता कोन माने हमें 5 prime रो हमें गोटे strand this is the sense strand this is the antisense strand हमें sense strand रो हमें जदी left to right को पड़ी पड़ी जिबा हमें कोन पहुचे AAGCTT. Okay, now एबे antisense strand रो हमें 5 prime रो 3 prime को पड़ीबा तारो ठीक तारो कॉम्प्लीमेंटरी बेस उडा को हमें 5 प्राइम रो 3 प्राइम को पढिबा हमें कोन पढुचे सेही सेम ता को पढुचे AAGCT सेही सेम सीक्वेंस ता को हमें पढुचे सो किंतु सेही सीक्वेंस ता से दीटा जाको सीक्वेंस कोन लेफ्ट टू राइट रे ही हमें पढुचे कि नाही दीटा जाको सीक्वेंस सेम अछंती केते बेले सेम अछंती जेते बेले गोटे सीक्वेंस हमें लेफ्ट टू राइट रो पढुचे आ गोटे सीक्वेंस राइट टू लेफ्ट रो पढुचे सो दैट इज द palindromic sequence. Okay, and this is about 4 to 6 proti restriction enzyme ro palindromic sequence ta 4 or 6 base per long. Okay, so this is the sequence for your HIN3. So आमे छुआ मानन को कहि नेबा दरकार बेले बेले हमर ए जो restrictions palindromic बा restriction site थाए किछिटा general restriction site रो sequence उडा को भी से माने मने रखि नेबे कारण किछिटा entrance उडा करे से माने जेमिति कि HIN3 ECOR1 ए दीटा जाको बहुत कॉमन सो ए दीटा रो से माने निहाती restriction साइट ता से माने माइंड रे रखि नेथिबे बेले बेले एटा मध्य आसे देन Swiss microbiology Werner Haber and Stuart Lin discovered restriction enzyme amara restriction enzyme generally it is I am not talking about HIN3 कि first restriction enzyme discover करथिले this question is also there in the multiple choice question it is Werner Haber and Stuart Lin. then which was the discover हेतिला किंतु isolate first कोनटा हेतिला isolate को restriction enzyme हेतिला discovery और isolation भितरे बहुत तफात अछि first discovery है isolation माने आमे सेटा को से बैक्टीरिया रु आमे बाहर को बाहर करछे आ आमे आम पर्पस पई यूज करछे आमे आमे आम जेनेटिक इंजीनियरिंग का बा रिकॉम्बिनेंट डीएनए पे आमे से एंजाइम टा को यूज करते सो फर्स्ट कोटा आइसोलेट हेतला फर्स्ट रेस्ट्रिक्शन एंजाइम टू बी आइसोलेटेड इज हिंट्री एटा क्वेश्चन राची एंड हु आइसोलेटेड इट वाज हैमिल्टन स्मिथ इन 1970 हैमिल्टन स्मिथ 1970 रे से आइसोलेट करथिले तापरे रेस्ट्रिक्शन एंजाइम कट एट रेस्ट्रिक्शन साइट्स दोस आर डैश palindromic. Let us again go to this questions. So we can see the enzymes commonly used in genetic engineering are restriction endonuclease खाली endonuclease नहीं restriction endonuclease और ligase हमको ligase ता परे हमको combine करवा पे दरकार so इटा restriction endonuclease और ligase polymerase ता हमें हमरो replication पे दरकार पॉलीमरेज का रेप्लिकेशन पाई दरकार किबा हमें पीसीआर जते बेले कहउचे सेति पाई दरकार किंतु जनरल रिकॉम्बिनेंट डीएनए हमें जते बेले करूचे गे बा जेनेटिक इंजीनियरिंग कहउचे इट इज ऑलवेज द रेस्ट्रिक्शन एंडोन्यूक्लियस एंड लाइगेज नॉट द एंडोन्यूक्लियस एंड लाइगेज सो एटा टिके छुआ मानन का पाई कंफ्यूजिंग हे पारे एठी देखंतु मॉलिक्यूलस इज अ केमिकल स्कैल्पेल रेस्ट्रिक्शन एंजाइम देन enzymes that cut the DNA molecule into fragment with sticky ends. It is a restriction enzyme or restriction endonuclease. Sticky end माने कौन आमे टिके देखी ना बा एठी देखंतु हिंट 3 जेते बेले कट करची हिंट 3 ए जो फर्स्ट 5 प्राइम रो आमे जेते बेले आछे 5 प्राइम रो दीटा जो ए अछि से ए दीटा ए रो फॉस्फोडाइस्टर बॉन्ड टाकु ही लीव करबो देन एपटिया साइड रो एंटाइसन स्ट्रैंड रो बी फर्स्ट दीटा ए रो फॉस्फोडाइस्टर बॉन्ड रे क्लीव करबो बा काटिबो तो हमको जो रिजल्टेंट डीएनए जो पिंक कलर रे जो आपण देखु चंती दैट इज अ रेस्ट्रिक्शन हिंट 3 रेस्ट्रिक्शन एंजाइम सो हमको जो डीएनए टा जो मिलिबो हम जदी फॉस्फोडाइस्टर बॉन्ड कटि गला ए भितरे तो हमर जो डबल बॉन्ड और ट्रिपल बॉन्ड जो बेसेस उडा का भितरे अछि सेई जाको आराम से भांगी जाय से जाको आराम से भांगी जाए सो से गुडा को भांगी जानती आ हमको एमिति स्टिकी एंड मिले एटा को हमें काहे के स्टिकी एंड कहूचे कारण एटा को पुनी जेते हमें लाइगेस एंजाइम देकी ए दीटा जाको स्ट्रैंड को हमें जेते बेले ऐड करचे से माने फर्स्ट एनिल हे जानती ए जो कॉम्प्लीमेंटरी बेस पेयर रे एनिल हे जानती आ लाइगेशन पाई बहुत सुविधा हुए सेति पाई हमें स्टिकी एंड कहूचे स्टिकी एंड परे आउ गोटे भी कट अछि so this is a sticky end cut overhanging amara roche eta hoche sticky end आउ भी किंतु गुडाय रेस्ट्रिक्शन एंजाइम अछंती जो माने कि स्टिकी एंड दियंतिनी जो माने कि ठीक मझिरु कट कर दियंती एठी देखंतु एठी टी आउ सी मझिरे से कट करची आ तळटा भी जी आउ ए मझिरे से कट करची ओके सो हमर एठी किछि स्टिकी एंड किछि आ आसीनी से रेस्ट्रिक्शन एंजाइम ता जो प्रोड्यूस करछि किछि स्टिकी एंड आ आसीनी हमको गोटे ब्लंट एंड एटा को हमें कहबा ब्लंट एंड एटा को हमें कहबा स्टिकी एंड सो एटा किछिटा एग्जांपल हिंट 3 प्रोट्रूडिंग एंड किबा स्टिकी एंड हमें एटा को प्रोट्रूडिंग एंड भी कहउछे कारण किछिटा सीक्वेंस बाहर को डबल स्ट्रैंडेड डीएनए रो किछिटा सिंगल स्ट्रैंडेड बेसेस बाहर की अछंती पीएसटी भी आमर सेमिति प्रोट्रूडिंग एंड दिए किंतु इको आरवी आमर ब्लंट एंड दिए से टी आउ ए आउ टी मझिरे काटे आ हमको ब्लंट एंड दिए then let us go back to this one again then it enzyme used for opening up the cell for genetic experiment मु कहितली लाइसिंग एंजाइम लाइसोजाइम भितरे भी मु कहिची then which among the enzyme are cleaving enzyme which among these enzyme are cleaving enzyme exonuclease endonuclease restriction endonuclease आमे cleaving enzyme जेतबे पढिले आमे देखिले all these three are cleaving enzyme ए माने डीएनए को काटंती so ए सब तक all the above होबो answer then restriction endonuclease are utilized in genetic engineering as molecular buildup at molecular buildup at nucleotide, molecular degradation to DNA breakup, molecular scalpel for cutting DNA bits into long chain, and molecular cement for combining DNA bits into long chain. So basically restrictive enzyme ta amara molecular scalpel. Sadla vele restriction enzyme are molecular scalpel and it is used for cutting DNA. आमे long ta no ye करके आमे खाली एटा भी आमे लेखि दे परिबा. It is used for cutting the DNA. Okay. Then restriction endonuclease. एटा restriction enzyme रो जेते different types of question हे परिबा एठी मु include करी नेछि. Then restriction endonuclease are present in mammalian cell for degradation of DNA? No, we do not have that. They are used in genetic engineering for ligating to DNA. आमे ligation आमे दीटा gDNA को जोड़िबा पे आमे केबे भी restriction endonuclease नोने. Are used for in vitro DNA synthesis? नाइ आमर in vitro DNA synthesis पे आमर केबे भी आमे restriction endonuclease नोने. आमे polymerase नहीं पारिबा नेबा किंतु आमे restriction endonuclease नोने. Then what is the correct answer? Are synthesized by bacteria as part of their defense system. It is synthesized by bacteria as part of their defense system. Bacteria mananka bhitere restriction endonuclease kahinki achi. Bacteria mananka bhitere restriction enzyme eithipai achi because it is a part of it helps in their defense. Tanku gote amara jemiti immune system achi a restriction enzyme udaka semananka defense mechanism. Okay, these are bacterial protein. So amara gote common mind ko ase. This restriction enzyme they cleave DNA. तो bacteria रो भी DNA अछि एमाने bacteria रो भी DNA टा को काटी देई परिबे नाही एमाने चिन्हंती bacterial DNA कोटा आउ foreign DNA कोटा केमिति चिन्हंती कारण bacterial DNA टा methylated गोटे methyl group is added. There is an enzyme which is known as a methylase, and this methylase adds your methyl group to particular bases of the DNA of bacteria. So methylated कोटी methylated होता है सबु restriction sites उड़ा करे methylated होता है. Bacteria भीतरे जेते restriction site अछि जेते restriction enzyme पे जेते restriction sites उड़ा को अच्छंती से प्रति restriction site रे methyl group add होता है. So methyl group add हला माने से restriction enzyme टा आउ ए sequence टा को चिन्ही परिबन एटा eco R1 रो sequence किंतु जेतेबेले गोटे मिथाइल ग्रुप ऐड हे जाय से एंजाइम टा डीएनए ऊपर आसीकी बसी परिबन बिकॉज़ इट इज स्ट्रक्चर स्पेसिफिक. इट इज स्ट्रक्चर स्पेसिफिक से खाली डबल स्ट्रैंडेड डीएनए अनमिथाइलेटेड एठी जेमिति अछि एठी आसीकी बसी परिबन किंतु मिथाइलेटेड रे बसी परिबन. बैक्टीरिया रो डीएनए टा इट इज मिथाइलेटेड दस ए रेस्ट्रिक्शन एंजाइम उडा को ताकू किछि करंतनी किंतु बाहरु जो भी डीएनए आसला बैक्टीरियोफाज bacteria infecting virus उड़ा को bacteriophage को आवे. Suppose a virus bacteriophage आसी ता DNA टा को bacteria भीतरे insert कर दला. Then what happens is this restriction enzymes they cut the DNA. They cut the DNA because this is a foreign DNA and it will be creating problem to the bacteria. Thus it cut the DNA. So this restriction enzyme and methylase they together constitute the restriction modification system and eta tara defense mechanism re use hue bacteria ro defense mechanism re use hue. Okay. So this restriction endonuclease udaka bacteria they are bacterial protein now defense mechanism re use hue. Restriction enzyme are used in genetic engineering because they can cut a specific they can cut the DNA at a specific base sequence. They can खाली भी लेखाई थाई पारे they can cut a DNA. से माने खाली DNA टा को cut करंतिनी से माने DNA टा को specific base sequence रे cut करंति. Then which of the following enzyme in bacteria are responsible for restricting the growth of the virus? Bacteria भितरे virus रो growth टा को restrict करिबा पे हमें कोटा use करछे. This is restriction endonuclease. Okay, so again restriction endonuclease रो मु जो कहितली discovered किए discover करथिले then Verneuve and Swartland. first restriction endonuclease was isolated by Hamilton Smith. এই জায়গা আমি ডিসকাস করছি and we had included. First restriction enzyme isolated was Hin3. Hin3 amar isolated hitla in 1970. Restriction site have DNA sequence that are palindromic. Eti mo palindromic sequence kono pilla mananko understanding pai, if you are circulating this among students, so for their understanding, a bit of their understanding, it is palindromic sequence kono mu add kari nechi. Eco R1 exhibits a two-fold rotational symmetry. Two-fold rotational symmetry is related to this palindromic sequence. It is palindromic sequence and there are two-fold rotational symmetry also. So it is 2. Eco R1 ta kono? Eco R1 ta gote restriction enzyme. Then restriction modification system of bacteria involved. Ame ebe just restriction modification system ame dekhitile, to seta kon involve kochi restriction endonuclease and methylase. And also I've added a bit of note from my slide only. So either a student can directly go to the slide and find each and everything, or एथुरु भी सेमाने देखि कि स्लाइड को ये करके भी क्वेश्चन आंसर उडा का सेमाने आराम से आंसर करनी पारिबे. The DNA is genetically methylated. मु एबे methylation ऊपर कहितली हमर bacterial DNA टा methylate हुए, so say is genetically methylated at C base. हमर C base रे DNA टा methylated mostly amara C base re DNA ta methylated हुए. Then bacteria protect themselves from virus by fragmenting viral DNA with the help of एठी देखंतु एठी restriction endonuclease की किछि भी देइन आउ एठी देछि DNA हमें जानि ने एटा it is a linear DNA or a circular DNA. So आमे दीटा जाको भाबी नेबा जदि सर्कुलर आमे दीटा जाको DNA लिनियर DNA और सर्कुलर DNA को बोथ किए कर परिबो, इट इज द एंडोन्यूक्लियस. एक्सोन्यूक्लियस कोन खाली ओपन DNA टा को काटी परिबो. ओके, सो एठी पुनी से देछि बैक्टीरिया प्रोटेक्ट देमसेल्व्स फ्रॉम वायरस. बैक्टीरिया विषय कहिछि, सो बैक्टीरिया विषय जदि कहिछि से रेस्ट्रिक्शन न भी देखिले, इट इज ऑलवेज द एंडोन्यूक्लियस. It is always the endonucleus. Then, there's a bacterial protein for cleaving foreign DNA. Again, say question, say restriction endonucleus ro say part ta ko सबु जगह रे अलग अलग वे रे पचरा हैछि. So bacterial protein जोटा कि DNA को क्लीव करे आउ तार डिफेंस मैकेनिज्म रे हेल्प करे, it is the restriction endonucleus. Then which of the following enzyme is used to join bits of DNA? ए भितरु डीएनए को जॉइन करिवारे दीटा डीएनए फ्रैगमेंट को किए जॉइन कर पारे, इट इज ऑलवेज द लाइगेस. हमें ऑलरेडी डील कर सालेनी. So we have already dealt is ligase, DNA ligase. This is an enzyme that forms the phosphodiester bond at the end of the DNA strand that are already held together by base pairing of two extensors. आमे आगुरु देखितिले जेते बेले restriction endonuclease कट करे से गोटे sticky end protruding end रे कट करे. DNA ligase टा ए दूर रे थिबा दीटा protruding and DNA को सेमाने जाई कि लाइगेट करि परिबेनी बा फॉस्फोडाइएस्टर बॉन्ड तयारी करि परिबेनी. सेमाने केते बेले करिवे जेते बेले कि ए जो कॉम्प्लीमेंटरी बेस उडा को जोडी हे जायछन. आगो कॉम्प्लीमेंटरी बेस पेयरिंग हेइ जिवो, तापरे ए एंड ता जो रहिला एथिरे हमर फॉस्फोडाइएस्टर बॉन्ड ता तयारी हो. आउ गोटे भी क्वेश्चन आसी पारे, phosphate terminal phosphate group can be removed by which enzyme? Terminal एटा हमर जेते बेले restriction enzyme जेते बेले कट करे हमर गोटे phosphate group ते थाए. हमर गोटे phosphate o group हमर रहि जाई थाए. So ए phosphate group जदि रहिला हमर ligation हेई पारिबो नाही. हमर ligation हेबा पे यूजुअली हमें कोन करू, आफ्टर restriction enzyme cause the plasmid we add alkaline phosphatase जे कि ए phosphate group ता को काढी दबो. हला, so phosphate group ता को काढिबा एटा होची terminal phosphate group ता को काढिबा. सो एटा को काढिबा पे आमे अल्कलाइन फॉस्फेटेस आमे यूज करि थाउ अल्कलाइन फॉस्फेटेस okay then the site at DNA where restriction enzyme generally acts is a palindromic sequence is a palindromic sequence 10 dim repeats achi CG reach achi Tata box achi no it will always act at the palindromic sequence then which of the following enzyme is necessary to make accurate copy of a vector DNA accurate copies of a vector DNA आमर वेक्टर डीएनए रो एक्यूरेट कॉपी हेबा पे आमर गोटे एंडोन्यूक्लियस आमर दरकार आमर गोटे एंडोन्यूक्लियस आमर दरकार काहे की कारण आपण कहि परिबे माने डीएनए पॉलीमरेज एक्यूरेट कॉपी करि परिबो किंतु एटा जदी आमर रिकॉम्बिनेंट डीएनए आमे जदी कहूचे ताले रिकॉम्बिनेंट डीएनए रे आगो हमकु गोटे एंडोन्यूक्लियस दरकार जे की सेम पोजीशन प्रतिटी वेक्टर रो सेम पोजीशन रे से कट करिवो आमर जदी गोटे एंडोन्यूक्लियस अलग अलग पोजीशन रे कट करिकि आमे इंसर्ट करथिबा आमे एक्यूरेट वेक्टर आमे केबे भी पाई परिबा नै सो आमर बेस आमर फर्स्ट जोटा आमे गोटे एक्यूरेट वेक्टर एक्यूरेट कॉपी हमकु जोटा दरकार इट इज द रेस्ट्रिक्शन इट इज द एंडोन्यूक्लियस ओके देन in recombinant DNA technology, a plasmid vector must be cleaved by the same enzyme that has cleaved the donor gene. Yaro answer hochi a plasmid vector must be cleaved by the same enzyme that has cleaved the donor gene. आउ अलग ये गुडा कभी अच्छी मॉडिफाइड डीएनए लाइगेज अल्कलाइन फॉस्फेटेस डीएनए पॉलीमरेज ये गुडा कभी अच्छी बट द करेक्ट आंसर इज दिस वन व्हाई बिकॉज this is suppose the vector DNA and this is the any gene of interest or the foreign DNA आमर वेक्टर आमे फॉरेन डीएनए माने आमर जीन ऑफ इंटरेस्ट आमे से जीन ऑफ इंटरेस्ट गोटे ऑर्गेनिज्म रो जीनोम रो बाहर करबा पे गोटे सेल भीतर बाहर करबा पे आमे गोटे प्रकार रेस्ट्रिक्शन एंडोन्यूक्लियस यूज करछे व्हिच इज इको R1 हला सो इको R1 रे मु जेतळे काटिली मोर स्टिकी एंड आसला AAPG मोर स्टिकी एंड जोटा आसिला माने जोटा की कॉम्प्लीमेंटरी तारो बेस पेयर आ नाही खाली फ्री रे सिंगल डीएनए टा अछि तापरे डबल हेलिकल डीएनए टा अछि सो फ्री रे जोटा अछि AAPG ओके मु कोन करबी मु मोर डीएनए जो जा भितरे कि मु एटा को इंट्रोड्यूस करबा पे चाहूची बा लाइगेट करबा पे चाहूची सेही डीएनए टा को मु से सेम इको R1 रे ही मु काटिबी काहे के काटिबी मु जदी सपोज हिंट 3 रे काटुची मो प्रोट्रूडिंग एंड टा कोन हला TCGA मोर एठी कॉम्प्लीमेंटरी बेस पेयर करि परिबनी मो डीएनए सहित सेथि पय हमर बेंचमार्क होची हमें जते बेले रिकॉम्बिनेंट डीएनए हमें जते बेले प्रोड्यूस करछे हमर जीन ऑफ इंटरेस्ट आ हमर वेक्टर टा को हमें सेम रेस्ट्रिक्शन एंजाइम रे काटुचे ताकि ए जो जीन ऑफ इंटरेस्ट कोन करबो हमर वेक्टर रो ओवरहैंगिंग सहित कॉम्प्लीमेंटरी बेस पेयरिंग आगो करि परिबो तापरे डीएनए लाइगेस आसीकी एक्सक्यूज मी सेटा को आसीकी लाइगेट करि परिबो सो दिस इज द बेसिक रिक्वायरमेंट then which of the following sequence along a double stranded DNA molecule can be recognized as a cutting site it is a site of sorry as a cutting site for a particular restriction enzyme गोटे पर्टिकुलर रेस्ट्रिक्शन एंजाइम पय कटिंग साइट हमें कोटा को एटा गोटे एटा जो अलग अलग देइची एटा फॉरवर्ड सीक्वेंस एटा रिवर्स सीक्वेंस आमे सेंस एंटाइसेंस रे आमे फर्स्ट जदी देखिबा AAGG तापरे TTCC AAGG ता TTCC सहित आसी कि बेस पेयर कर परिबन TTC एटी TT A सहित T ता सॉरी सी एपटू जिबा हां ATGC 80 so if we are going so this is correct मु एटा को खाली टिके चेंज कर दिए mu change kar diye, maybe I'll include G. I'll include G. Okay. So ebe alaga hai gala A G G सहित A T टा हमर बेस पेयर कला C टा बेस पेयर करिवो कॉम्प्लीमेंटरी बेस पेयर करिवो किंतु G G सहित कॉम्प्लीमेंटरी बेस पेयर हे पारिबो नाही. Then A G T C. Just a minute, what is recognized as a which of the sequence double stranded DNA can be recognized as a cutting site? Cutting site. Then AGTC. So AGTC. T टा करिबो एटा भी कटिंग साइट हे परिबनी एटा भी मु सी करे एटा भी मु अलगा टिके कर दिए एटा भी मु ए कर दिए. ओके. सो एठी हमें जदी देखिबा मु एटा एक्चुअली सबु तक मु पैलेंड्रोम मु देई देथिली एटा हमें जदी देखिबा हमर AGTC रे हमर TCA एटा आसी कि हमर एनिल कर परिबो किंतु CC एनिल कर परिबन. सो एटा पैलेंड्रोमिक टी सीक्वेंस हे परिबन. देन GGCC मु जदी लेफ्ट टू राइट पढिबी एटा भी GGCC होउची. राइट टू लेफ्ट पढिली GGCC, लेफ्ट टू राइट पढिली GGCC. So this is a palindromic sequence. So confusion ना हबा पे हमें छुआ मानु को कहिबा सपोज गोटे पैलेंड्रोम हमको एमिति दिया हेतिबो हमें एपटू फर्स्ट राइट टू लेफ्ट फर्स्ट को हमें राइट टू लेफ्ट पढिबा तापटा सॉरी लेफ्ट टू राइट पढिबा फर्स्ट तापटा राइट टू लेफ्ट पढिबा. सो जदी हमर GGCC गोटे सीक्वेंस हला एटा राइट टू लेफ्ट पल्ला बेले भी हमें सेही सीक्वेंस ता GGCC पहुचे. सो दैट इज अ पैलेंड्रोम सीक्वेंस. सो पैलेंड्रोम सीक्वेंस हला माने ही हमर रेस्ट्रिक्शन एंजाइम ता रिकॉग्नाइज करि पारिबो. सो दिस इज अबाउट एमितिका आउरी किछि भी माने हमें से मानं को एग्जांपल दे देले से मानकर सुविधा हे जिवो फॉर एग्जाम पॉइंट ऑफ व्यू. देन एग्जांपल ऑफ टू सिंथेसाइजिंग एंजाइम हमर क्लीविंग एंजाइम हे गला एबे हमें आसीबा सिंथेसाइजिंग एंजाइम को देटा एंजाइम सिंथेसिस करिबारे हमको हेल्प करे. सिंथेसिस माने तयारी करिबारे. इट इज ऑलवेज डीएनए पॉलीमरेज एंड रिवर्स transcriptase. So let us see what is this DNA polymerase and reverse transcriptase. So over here we can see these are the synthesizing enzymes. DNA polymerase, this helps in the synthesis of complementary DNA strand on a DNA template. एटा देखंतु एटा गोटे DNA template. एटा गोटे DNA template. एटा हमर डीएनए सिंथेसिस पई सदा बेले गोटे शॉर्ट प्राइमर हमकु दरकार. प्राइमर माने कोन अराउंड 20 रु 30 बेस पेयर बेले बेले हमें 15 बेस पेयर बिनो 20 रु 30 बेस पेयर डीएनए फ्रैगमेंट जोटा कि एग्जैक्टली कॉम्प्लीमेंटरी टू द इनिशियल पोर्शन ऑफ द डीएनए जोटा कि एक्सटेंशन जोटा को कि हमें तयारी करबा को जाउछे. सो दैट इज द प्राइमर. इवन ड्यूरिंग रेप्लिकेशन आल्सो वी रिक्वायर प्राइमर. सो प्राइमर आसी के फर्स्ट रे एनिल करिगला इनिशियल सीक्वेंस को एनिल कला प्राइमर एनिल कला परे ही हमर पॉलीमरेज ता आसी के बाइंड करबो तापरे पॉलीमरेज गोटे गोटे कॉम्प्लीमेंटरी बेस ऐड करी करी जिवो आ हमर एसो हमर गोटे सिंगल स्ट्रैंडेड डीएनए ऊपर हमर डबल स्ट्रैंडेड डीएनए तयारी हे गला. देन दिस पॉलीमरेज विल डिसोसिएट. Then what is the reverse transcriptase? Is help in the synthesis of complementary DNA on a RNA template. DNA polymerase DNA template रे ही DNA तयारी करेला किंतु RNA polymerase ता कोन करबो हमरो. Let me give an example. हमरो complementary reverse transcriptase ता हमरो mRNA अछि mRNA को बाइंड करिवो आ mRNA ऊपरे हमर DNA तयारी करिवो. mRNA, this is the mRNA. What is the indication of mRNA? It is having uracil. हमर mRNA रे uracil थाए किंतु से uracil ता हमर DNA रे that is removed and another base which is T is added. So गोटे mRNA रे जेते बेले आसीबो रिवर्स ट्रांसक्रिप्टेज आसीबो से गोटे डीएनए तयारी करिवो. ए जगह रे टी हला यू जगह रे सदा बेले ए रहलो काहे के सेटा टी रो टी रो मॉडिफिकेशन ता सेटा मॉडिफाइड बेस ता सो यू जगह रे सदा बेले ए रहबो सो से एमिति गोटे डीएनए तयारी करी वाला सो दिस इज नोन एज अ cDNA c ता स्मॉल रे रहबो व्हिच वी कॉल एज अ कॉम्प्लीमेंटरी डीएनए अगेन आमे जते बेले जिबा क्वेश्चन आंसर को आमे जते बेले जिबा सेथिरे रिवर्स ट्रांसक्रिप्टेज विषय रे भी अछि who discovered reverse transcriptase it was Temin and Baltimore in 1970 Temin and Baltimore in 1970 this is a very common question so what is a template for reverse transcriptase template kono it is always the mRNA ame RNA lekhi dele bi pilla mane ठीक हबनी हमर सदा बेले messenger RNA ऊपर ही हमर reverse transcriptase bind करके तार DNA टी को से तयारी करी थाए so now let us go to the another important feature of this cDNA is that it does not have intron आमर प्रोकैरियोट रे आमर इंट्रॉन नहांती आमर प्रोकैरियोट रे आरंभ रु एंड पर्यंत खाली स्ट्रक्चरल जीन अछंती स्ट्रक्चरल जीन माने सपोज एडी पांचटा जीन अछंती पांचटा जाको जीन भितरे किछि भी गैप नै आ से पांचटा उडा को जीन माने कोन gene is not it is not referring to any DNA gene is that region of the DNA that actually codes for a mRNA जो region of DNA जो region of DNA inside the chromosome codes for a mRNA that is known as a gene okay so functional DNA भी हमें कही था से mRNA करेला mRNA टा प्रोटीन करेला हमरो प्रोकैरियोट्स रे किछि भी जीन गुडा का भितरे किछि गोटे सपोज जीन अछि स्ट्रक्चरल गोटे जीन भितरे किछि भी गैप नै suppose this is a for example this is a insulin gene for example मु खाली एग्जांपल नोची एटा फर्स्ट टू एंड पर्यंत तार सीक्वेंस ता जो अछि खाली इंसुलिन जीन रो ही सीक्वेंस अछि किंतु यूकैरियोट से हमर कोन होछि हमर इंट्रॉन हमर यूकैरियोट रो जीन उडा को हमें स्प्लिट जीन कहूचे we are having introns in between okay so this 1 2 3 4 5 these are the exon exon ए जो 1 2 3 4 5 हमें जदी मिसे देले ताहले हमको गोटे फंक्शनल जीन मिलिबो हला सो ए फंक्शनल सॉरी फंक्शनल mRNA हमको मिलिबो सॉरी फंक्शनल mRNA हमको मिलिबो सो ए फंक्शनल mRNA पाइबा पय हमर कोन होची हमर जो ट्रांसक्रिप्शन जो होची हमर गोटे RNA प्राइमरी ट्रांसक्रिप्ट RNA रो प्राइमरी ट्रांसक्रिप्ट होची प्रोकैरियोट्स रे डायरेक्टली mRNA सिंथेसिस होची प्रोकैरियोट रे जेते बेले RNA पॉलीमरेज ट्रांसक्रिप्शन करची डायरेक्टली mRNA से तयारी करची किंतु यूकैरियोट रे इंट्रॉन सहित गोटे RNA टे तयारी होची सेटा mRNA नै सेटा प्राइमरी ट्रांसक्रिप्ट तापरे हमर RNA प्रोसेसिंग मशीनरी अछि यूकैरियोट्स भितरे RNA प्रोसेसिंग मशीनरी अछि से माने ई इंट्रॉन्स बा हमें जाको जंक DNA भी कहू हला हमर एवडा दरकार नै हमको खाली ब्लू उडा का दरकार कारण ए ब्लू रीजन उडा कहीं इंसुलिन प्रोटीन को कोड करचंती सो ए इंट्रॉन उडा को से माने काढी देकी एक्सॉन उडा को जोड़ी दोचंती सो दिस इज द फंक्शनल ट्रांसक्रिप्ट ऑफ mRNA सो यूकैरियोट रे केबे भी डायरेक्टली mRNA हो आगो गोटे प्राइमरी ट्रांसक्रिप्ट होची तापरे mRNA सो हमर जदी mRNA रो जदी हमर DNA टा होची सपोज एटा mRNA या ऊपर गोटे DNA जदी सिंथेसाइज हला ता माने कोन हमर इंट्रॉन उडा को नाही जदी इंट्रॉन उडा को नाही एई जो DNA जो सिंथेसाइज हला एटा मु यूकैरियोट्स सॉरी प्रोकैरियोट्स भितरे भी मु एक्सप्रेशन पई यूज करि पारिबी किंतु नॉर्मली मु यूकैरियोटिक जीन प्रोकैरियोट रे इंट्रोड्यूस करि पारिबिनी काहे कि कारण तांकर आरएनए प्रोसेसिंग मशीनरी नाही काहे कि तांकर दरकार नाही से मानक स्ट्रक्चरल जीन कंप्लीट स्ट्रक्चरल जीन अछि विदाउट एनी इंटरप्शन ओके सो मु जदी mRNA रो cDNA तयारी करछि आई कैन यूज दैट cDNA फॉर एक्सप्रेशन ऑफ माय जीन ऑफ इंटरेस्ट इन प्रोकैरियोट्स let us go to the question again. Okay. So our synthesizing enzymes are DNA polymerase or reverse transcriptase. Then DNA ligase catalyzes the formation of phosphodiester bond. Glycosidic carbohydrate ro peptide bond ta protein ro amino acid jodiba pe. So it is phosphodiester bond. The gene carried by recombinant DNA is known as recombinant DNA भितरे जो gene टा जो carry करा है gene of interest सेटा को हमें transgene कहूचे. Let me see. एठी देखंतु एटा गोटे vector हला. So this is the gene of interest. This is suppose this is the gene of interest. So I'll be calling this gene of vector को हमें केबे भी transgene कहिबानी. Vector is vector. It is just used for transferring. It is just used for transferring this gene from suppose human into a bacteria. ताले हमर बैक्टीरिया भीतर हमर ट्रांसफर करबा काहे की दरकार हमर तो सिंथेसाइज होची हमर हमें जानि छे इंसुलिन मोस्ट ऑफ अस आर डायबिटिक सो आमे बैक्टीरिया भीतर जदी एटा को एक्सप्रेशन करके बहुत अधिक इंसुलिन आमे जदी प्रोड्यूस कर पाले बैक्टीरिया भीतर ही काहे की आमे इंट्रोड्यूस बैक्टीरिया भीतर ही मु कहबिनी आरो भी गुडाय प्रोकैरियोट्स भीतर आमे इंट्रोड्यूस कर परिबा किंतु लोअर ऑर्गेनिज्म भीतर आमे काहे की इंट्रोड्यूस करछे कारण से माने बहुत जल्दी डिवाइड करंती आ सेहिति पय जेहेतु बहुत जल्दी डिवाइड करंती द अमाउंट ऑफ प्रोडक्ट व्हिच इज प्रोड्यूस्ड इन अ सिंगल डे वी कांट इमेजिन वी कैन नेवर प्रोड्यूस दैट मच. So it is to get a bulk of the product recombinant protein bulk recombinant protein पाइबा पई आमे एटा आमे यूज करछे. Okay, then let me go again to the okay, so it is known as a transgene. Then DNA molecule used for carrying the foreign DNA एटी foreign DNA जगह रे transgene भी कहि पारंती और gene of interest भी कहि पारंती और DNA of interest to be cloned भी लिखा जाय पारे is known as आमे कहा भीतर carrier DNA टा किए DNA molecule used for carrying the foreign DNA. It is the vector. It is the vector. Okay. Then recombinant DNA method. In recombinant DNA method, the term vector refers to vector or definition. A plasmid or other agent used to transfer DNA into living cell. It is a carrier. So एटा plasmid हे पारे और other agent आउ किछि आमे plasmid बादे आमे bacteriophage भी यूज करू आमे yeast artificial chromosome यूज करू bacterial human artificial chromosome भी आमे यूज करता. So plasmid बादे अन्य किछि भी agent जोटा कि हमरो gene of DNA of interest को transfer करिवारे help करे that is known as a vector. Sticky DNA fragments vector नुहे enzymes that cut the DNA into restriction fragment vector नुहे और DNA probe तभी vector नुहे. So what is true for plasmid? Plasmid पाए एक चार्टा जाका भीतरो true कौन? Found in virus? No. Contain gene for vital activities? No. Part of nuclear chromosome? It is never a part of nuclear chromosome. They are free. They are independent. What is true? Widely used in gene transfer. Gene transfer करिबा पे use ए पार. हमरो first artificial cloning vector. हमको एठी एटा भी देई पारे. What is the first artificial cloning vector? The first artificial cloning vector is PBR322. Okay. So a PBR322 टा किए construct करथिले. The first artificial cloning vector was constructed by Bolivar and Rodriguez. It is Bolivar and Rodriguez. So now again coming to a vector upare hi question udaka. The first artificial cloning vector it is PBR322. Okay. Then PBR322 is a plasmid belonging to एटा हमरा first artificially तैयारी हेतिला किंतु कोटु नै कि तैयारी हेतिला आगुरु गोटे plasmid कहा भितरे थिला से एस्चुरिकिया कॉली भितरे गोटे प्लाज्मिड थिला से प्लाज्मिड टाको नै कि माने बॉलिवर और रॉड्रिगस गोटे आर्टिफिशियल प्लाज्मिड तैयारी करले. ओके आर्टिफिस तो यार ना टाइम इति काहे कि होची आगु को गले या मानक नेमिंग ऊपर भी क्वेश्चन आसी परिबो इन सब्जेक्टिव टाइप या ना PBR322 काहे कि होची. P stands for plasmid, BR Bolivar Rodriguez सेटिबाई कैपिटल जो साइंटिस्ट दि जण जो ये करछन 322 से माने प्लाज्मिड माने आमे जते बेले तयारी करथाउ आमे गुडाए सेट ऑफ एक्सपेरिमेंट आमे करिथाउ से गुडाए सेट ऑफ एक्सपेरिमेंट कला बेले आमे तांकर नंबरिंग नेमिंग उडा को भी देथाउ सपोज पीबीआर 1 2 3 4 एमिति से करी करी कि 322 देन 400 भी मे बी जाइतिबे 400 से सो से माने देखले जे एक्चुअली तांको तांकर जो आर्टिफिशियल चेंजेस तांको जहा दरकार ए पीबीआर 322 रे 322 जोटा से 322 एक्सपेरिमेंट नंबर टारे ही तांकर अछि सो thus it contains the p small p stand for plasmid, br stand for this Bolivar and Rodriguez, and 322 तांकर experiment co experiment बा co set रे से माने find करथिले so this belongs to this pbr 322 initially माने native plasmid ता से Escherichia coli से बाहर करथिले which of the following are characteristic of vector has origin of replication yes it does have a origin of replication कारण origin of replication नथिले से केबे भी replicate करी निजे replicate करी परिबो नै से गोटे organism ऊपर dependent रहइबो से organism रो replication जेबे हबो से सेबे ही replication कर परिबो किंतु निजर origin of replication थिले से आराम से निजे केते बेले भी replicate कर परिबो then replicate autonomously yes it replicate autonomously and it does not depend upon the host in which it is introduced has a selectable marker yes it has a selectable marker so a tinita jako is true for this vector okay I think 1 hour is already done right so because of this time restriction I'm unable to discuss this other MCQ type of question so shall we go to the long question or you want me to discuss a few more of this and then we'll go to the long question whatever is comfortable for you hello
Yes, madam.
We'll be discussing this or we'll go to the long question.
Yes, madam.
Okay, so I'll be providing this MCQ, all the MCQs and their answers. I'll be providing it. So let us go to the long question. I'll just take a 2 minutes break. I'll just take a 2 minutes break and I'll come back. Thank you so
madam.
much. Welcome back, colleagues. Now we will be discussing about the subjective question. So basically, the subjective question we can give it as a short type question, long type question also. So students should be prepared to answer the question according to the marks that have been given. So I have not differentiated this question into 1 mark, 2 mark, 3 mark. All of them I have given together. So first, the basic one, what are biotechnology? Elucidate the principles of biotechnology. Okay, so what are biotechnology? We can find it in this slide itself, which is available. I given it to, I said it with the institution. So biotechnology is again, there are many definitions to biotechnology. There are many definitions to biotechnology. So students are at freedom to write any definition they want to. But I felt this as a most proper one, so I have taken this one. They can write any other definition of biotechnology. So biotechnology is an industrial application of biochemistry, microbiology, chemical engineering, genetic engineering, etc. in order to make the best use of capabilities of microorganism, culture tissue, cell, etc. So best use of capabilities, what do we mean? Microorganism, it is having its own existence. It does a particular thing in own specific way. तो निज वे से काम करे किंतु आमे ताको चाहूँ जे नहीं से ए वे रे काम न करू से ए वे रे एमितिया वे रे काम करू जोटा कि मैनकाइंड पाई हेल्पफुल हबो मैनकाइंड पाई केबे भी हार्मफुल हबनी बट मैनकाइंड पाई केमिति हेल्पफुल हबो तो सेटा होउची बायोटेक्नोलॉजी सो मेन प्रिंसिपल्स ऑफ बायोटेक्नोलॉजी कोन the two main principles of biotechnology are genetic engineering or recombinant DNA technology and maintenance of a sterile or contamination free ambience. यार आंसर उड़ा को भी मु ए क्वेश्चन उड़ा को तळे रे मु आंसर उड़ा को भी मु देई नै छी तो का असुविधा जेमिते न हो I provided the answers to you. So genetic engineering, any manipulation of DNA or any change to the structure of the DNA is known as a genetic engineering. Recombinant DNA technology, when we are combining from two different sources, DNA from two different sources, it is known as a recombinant DNA technology. Already we have dealt that Stanley Cohen and Herbert Boyle in 1972, they produced the first recombinant DNA. Now, next most important one is, or the next principle is maintenance of a sterile ambience. आमे जेते बेले भी live organism ऊपर काम करूचे, may it be microorganism, may it be for the purpose of genetic engineering, biotechnology, whatever it is. The most important condition आमको आगो PCR सिखिबा आमको दरकार नहीं आमको आगो क्लोनिंग सिखिबा दरकार नहीं आमको फर्स्ट जोडा दरकार होची आमे केमति ओटा स्टेराइल कर एबे कोरोना द्वारा आमे तो गुडाय जिंस सिखि सारलेनी बट जेते बेले आमे गोटे रिसर्च लैब को आमे जेते बेले जाऊ जेनेटिक इंजीनियरिंग किबा रिकॉम्बिनेंट डीएनए करबा पे आमे आउरी अधिका कंटामिनेशन फ्री एनवायरनमेंट रे आमे काम करि थाई काहे कि कारण ए जिंस ए रिकॉम्बिनेंट डीएनए रो जो फाइनल प्रोडक्ट जो बाहर अल्टीमेटली इट इज गोइंग टू मैनकाइंड so if there is any contamination mankind will be suffering amara definition kon thila amara definition hoche for the benefit of the mankind we can't give any product to human being which will cause side effect to them. Okay, so these two are the two most important principles of biotechnology. Then what is recombinant DNA technology? Again, recombinant DNA technology ta ame first jota discuss kale taha rahi. This is a short question. So recombinant DNA technology already I have discussed. Then what is genetic engineering? So genetic engineering too, it is a short question that I have discussed. Any forceful alteration or manipulation that we are carrying out on DNA is known as a genetic engineering. Then who constructed the first recombinant DNA and how? Already we discussed Karsachi, Amara, Stanley Cohen and Herbert Boyle. they have discovered the first recombinant DNA, and how they have discovered, we need to discuss these. That is, first we have isolated the tetracycline R gene from Salmonella typhimurium. Then we are isolating plasmid from Escherichia coli. Then we are digesting the plasmid with restriction enzyme. We are opening up the plasmid. आमे plasmid टाको linear न करिबा पर्यंत आमे आम DNA टाको introduce करि परिबा ना. So restriction enzyme द्वारा आमे open कले. Then with the help of a ligase, we are ligating the tetracycline R gene into the plasmid. This is a recombinant molecule produced by Stanley Cohen and Herbert Bohr. This is a tetracycline resistant plasmid. This is a tetracycline resistant plasmid. Okay. Now, how list the tools used in recombinant DNA technology? So the tools are, I have also given in this PowerPoint. The tools are your enzyme. We can also, while writing, if this is a question that belong to maybe 3 or 4 marks, enzyme lekhi sari ki different type of enzymes, lysing enzyme, cleaving enzyme, synthesizing enzyme, DNA ligase, alkaline phosphatase, eguda ko bhi mane ame encourage kariba chua mananku lekhiba pai. Then recombinant DNA tiyari kariba pai vector insert amara darkar. Then we need a competent host, PCR, DNA probe, gene therapy. ए सब आमर जीन थेरेपी न लेखिकी भी आमे खाली restriction enzyme, polymerase, DNA polymerase, synthesizing enzyme एटी मु लेखिनी आमर synthesizing enzyme आमर दरकार. Then recombinant DNA, competent host, एतक आमर निहाती दरकार. आउ एटा हि साला परे जदि 5 मार्क रो क्वेश्चन अछि तो आमे एंजाइम गुडा को भी डिस्क्राइब करिबा. आउ जेमिति रेस्ट्रिक्शन एंजाइम को आमे डिस्क्राइब करिबा. देन पॉलीमरेज एंजाइम, सिंथेसाइजिंग एंजाइम टाको आमे टिके डिस्क्राइब कर देबा. देन व्हाट इज वेक्टर, व्हाट इज इंसर्ट, व्हाट इज अ कॉम्पिटेंट होस्ट. सो वी नीड टू गिव डेफिनेशन टू दैट आल्सो इफ इट इज अ लॉन्ग क्वेश्चन. डिपेंडिंग अपॉन द मार्क, स्टूडेंट्स शुड बी राइटिंग. देन okay, tool se gala pare what does eco R1 signify, how is it named here? A naming ta eco R1 ro kemiti heichi. So let us go to the naming. So nomenclature, how is this eco R1 or hint 3 amara eco R1 chhadi ki ama hint 3 bhi koi parodi. Ethi amara eco R1 koichi, so mu eco R1 ta ko nechi. E, always the first capitals, the first in capital letter always indicate genus of the particular species. Amara eco R1 uda ko restriction endonuclease or restriction enzyme uda ko amara bacteria ru asichi. तो को बैक्टीरिया रो से रेस्ट्रिक्शन एंजाइम से बैक्टीरिया रो जेनस ना फर्स्ट लेटर टा जोटा सेटा सदा बेले कैपिटल रे हमरा रेस्ट्रिक्शन एंजाइम रे फर्स्ट लेखा हुए. तापरे स्पीसीज कॉली रो सीओ बीटा फर्स्ट और सेकंड बीटा लेटर आमे इंक्लूड कले. देन दिस आर इज अ स्ट्रेन इको R1 one होची फर्स्ट ऑर्डर रे आमे जते बेले डिफरेंट सेट ऑफ रिएक्शन आमे बसो करू छे हमरा फर्स्ट ऑर्डर हम फर्स्ट रिएक्शन रे ही हमरा आसी जाई छी. सेति पाई सेटा इको R1. हिंट 3 रो हेमोफिलस इन्फ्लुएंजा रो आसी. सो फर्स्ट हमरा हिंट 3 केमि दियाला H कैपिटल genus name for hemophilus capital सेटा सदा बेले. देन influenza IN से दीटा सदा बेले स्मॉल रहला. RD होची तार स्ट्रेन ना सेति में हमर लास्ट टा नियाला D नियाला. देन 3 थर्ड ऑर्डर रे हमर एटा आइडेंटिफाई हेतिला. Okay, so this is how we will be naming. Then how are restriction endonuclease named? Again, how they are named? Write examples. ए दीटा जाको सॉरी ए दीटा जाको क्वेश्चन मोरलेस से एटी इको R1 दे देछि. एटी हमें किछि भी नहीं पारिबा, हिंट 3 नहीं पारिबा, PST नहीं पारिबा, इको R1 नहीं पारिबा, जहां भी नहीं पारिबा. then elucidate the steps in recombinant DNA technology. Amara recombinant DNA ର technology ର steps गुड़ाक भी आमे आमर एठी अछि. Just so steps of this recombinant DNA technology हम exactly अबे पाउनी ये टाको. Okay, so first we need to generation of DNA fragments. हमें first DNA fragment को generate कले हमारा required DNA fragment or the gene of interest. First we are isolating the gene of interest, then we are isolating the vector or the plasmid which will be carrying our gene of interest. Then we are digesting it with a restriction endonuclease, and we are then inserting the gene of interest into the vector and creating a recombinant DNA. Then after this we are introducing this one. The third step is introduction into host cell. After host cell माने जी हमारा ये recombinant DNA टाको ता भीतर को नहीं की ताको multiply तारो clones तैयारी कर पाई. सेटा होची host. After introduction into the host, we are selecting कौन सिलेक्ट करछे को होस्ट भितरे हमर रिकॉम्बिनेंट डीएनए अछि. बेले बेले कोन हुए खाली प्लाज्मिड ता भी चली जाई थाय. बेले बेले कोन हुए प्लाज्मिड ता कटिला परे रेस्ट्रिक्शन एंडोन्यूक्लियस काटिला परे आउथरे जेते बेले लाइगेज दिया हुए खाली प्लाज्मिड भी होस्ट भितर को चली जाई थाय. तो जो होस्ट भितरे खाली प्लाज्मिड अछि किबा किछि भी नाही हमें से मानं को डिस्कार्ड कर दोछु आ जहा भितरे खाली रिकॉम्बिनेंट डीएनए अछि हमें सेटा को हमें सिलेक्ट करके नै जाउछु. देयर इज अ स्क्रीनिंग प्रोसेस आल्सो. सो स्क्रीनिंग प्रोसेस रे हमें सिलेक्ट करलु, देन expression of the gene to produce the desired product. ए जो host भितरे हमर gene expression माने हमर ए gene ता mRNA form करिवो host भितरे और mRNA ता produce करिवो. So these are the basic steps in biotechnology, in the recombinant DNA technology. Okay, then define recombinant DNA. Already आमे एठी recombinant DNA technology आमे करिथिला आगु. एठी recombinant DNA माने a DNA which is DNA fragment having DNA from two different sources. A DNA fragment which is having DNA from two different sources are known as a recombinant DNA. What are nucleases? मो ऑलरेडी मो कहि साची जेते बेले हमें डील करथुले this MCQ type. So these are cutting both the DNA and the RNA. What are exonucleases? Already I have dealt with it. Then define restriction endonuclease and their role. Define restriction endonuclease and their role. So restriction endonuclease amaro we can define restriction endonuclease like these are the enzymes that are found only in bacteria and they are responsible for their defense mechanism. Okay, and this restriction enzyme helps the bacteria or it forms a part of restriction modification system also. And we are using this restriction enzyme for genetic engineering, as this restriction endonuclease cleaves the DNA at a particular site which is known as the restriction site. So एगुडाका आमे टिके माने restriction endonuclease कोन, bacteria रे तारो role कोन, आम पाई how it is helpful also we can mention it over here. Then elucidate the cleavage pattern of restriction endonuclease. I'm already discuss करछाती cleavage pattern ta. So I won't be going in एटी गोटे example entry नहीं कि मैं already discuss करछाती केमिति it's a cleave करछि and it is providing a sticky end. So this is how the students must be encouraged to even draw the diagram, even draw the से माने इमिति न करंतु जे entry रो sequence नहीं चंदी किंतु एटी इक्व r1 लिखि चंदी ना आमे सदा एनकरेज करबा जहार से माने सीक्वेंस नहीं चंदी से माने ताहार ही माने से पर्टिकुलर एंजाइम रही ना लिखिकी डायग्राम टी ड्रा करिवे आ केमिति पैलिंड्रोमिक सीक्वेंस रे काटुची बुझे कि तार स्टिकी एंड जो प्रोड्यूस करछि सेटा को भी मध्य से माने बुझेबे then what is restriction modification system? This has also been discussed. So restriction modification system ama bacteria re achi which is helping in its defense. So together this restriction endonuclease and methylase together is known as a restriction modification system. It is a defense mechanism. Methylase at methyl group at cysteine. एठी देखो मु गोटे नेइची गोटे पिक्चर नेइची वेब रू बट एठी से ए रे मिथाइलेट करछि जोटा कि भूल एठी से ए रे मिथाइलेट मु एमिति गोटे खाली एग्जांपल नहीं जाईची बट इट शुड ऑलवेज मिथाइलेट एट सी रे ही से मिथाइलेट करबा दरकार सो सी रे मिथाइलेशन कला मिथाइलेशन हला माने हमर बाहरु जो वायरस उडा को आछंती से माने अटैक करि परिबेनी डीएनए को एट द सेम टाइम हमर जो रेस्ट्रिक्शन एंडोन्यूक्लियस जो अछि बैक्टीरिया भितरे से मध्य निज डीएनए को से काटि परिबनी सो दिस मिथाइलेज and restriction endonuclease together is known as a restriction modification system. Then एटा मु already discuss कर साची मु एठी गुडाए जो क्वेश्चन उडा को देछि different pattern रा जहा आसी परिबा का देछि so sometimes it is repetition like what does H D and 3 refers in enzyme entry एमितिया क्वेश्चन भी आसी पारे. Then what are recognized sequence or recognition site? Recognized sequence or recognition site always deals with the restriction site of restriction endonuclease. हमारा restriction endonuclease रो restriction site से जो DNA रो जो site टा को recognize करे that is a recognizing sequence and that is known as a restriction site or the recognizing sequence. जेमिति कि एठी just a moment. This is the recognizing sequence or restriction site. This is always 4 to 6 base pair in long. So at the way say 4 to 6 base pair long time mostly on an average. Then how a sticky end forms on DNA? Why are they so formed? Why are this known as a आमे sticky end से गुड़ा को आमे काहे की कोहुचे कारण first, why are they formed? Sticky end are formed because the restriction endonuclease cleaves the DNA at palindromic site. से palindromic site रे cleave करे माने गोटे sequence रो 5 prime end रे जदी cleave कला A रो reverse sequence रे भी से right side रू आरंभ होई कि से A टा को cleave कला जेहेतु से palindromic site रे cleave कला आमा पाखरे किछिटा overhanging region अछि जोटा कि हमर माने कॉम्प्लीमेंटरी बेस उडा को कॉम्प्लीमेंटरी बेस पेयरिंग डिसॉल्व हो जाए आ हमर डीएनए टा पूरा कटि जाए हमको जो दीटा जो डीएनए मिलला तार किछिटा रीजन अछि जोटा कि सिंगल स्ट्रैंडेड बाकी ची बेस पेयर सिंगल स्ट्रैंडेड ही थाए ताको कॉम्प्लीमेंटरी डीएनए न थाए because of this it is known as a sticky ends so यार आंसर भी मु देई देइची माने तळे देई देइची मु खाली just I am discussing it I have written the answers too so it won't be a problem for anyone then so what would happen if restriction enzyme does not cut the DNA at specific site जदि आमर डीएनए टा नॉन स्पेसिफिकली आमर जदि कट कला ताहले आमे केबे भी क्लोनिंग सेडा रिकॉम्बिनेंट डीएनए टेक्नोलॉजी रे केबे भी आमे यूज करि पारिबानी कारण आमर लेट मी गिव एन एग्जांपल ओवर हियर सपोज आमर इको R1 टा कट कला से नॉन स्पेसिफिकली कट होची सपोज इंसर्ट टा को एटी एटीटी रे कट कला किंतु वेक्टर टा को ए भीतर आउ गोटे को रीजन रे कट कला सीसीजीजी रे से कट कला सो एठी जो आमर सीसीजीजी जो प्रोड्यूस हबो जो ये प्रोड्यूस हबो फ्री एंड जोटा आमर प्रोड्यूस हबो से एनिल कर परिबन से आमर एंजाइम्स से आमर जीन ऑफ इंटरेस्ट सहित एनिल करि परिबो सो आमको गोटे रिकॉम्बिनेंट डीएनए आमे केबे भी पाइबानी so it is always necessary that the restriction enzyme should always cut it आमे जेते थरा इको r1 पकेवा इको r1 सृष्टि प्रलय हे गले भी एई जो रेस्ट्रिक्शन साइट अछि एई जो सीक्वेंस अछि GGAATTC ए रेस्ट्रिक्शन साइट सराउ GGAATTC रो से खाली से G आउ ए भीतर ही एटी एरो दिया अछि G आउ ए भीतर ही कट करबो एपटु भी से G एपटु आसिले भी से G आउ ए भीतर ही से कट करबो से आउ कोठी भी कट करबो सो सदा बेले से G आउ ए भीतर ही कट करबो सेति पय हमें सदा बेले हमें सेटा को रिकॉम्बिनेंट डीएनए टेक्नोलॉजी रे हमें यूज करि नै पारिबा एठी गोटे एग्जांपल दिया छि जो ब्लू रे जो जाको जो दिस इज अ प्लाज्मिड और अ वेक्टर दिस इज the sticky end this is a sticky end जोटा कि हमें कट करके हमें this is gene of interest हमें बाहर करछे gene of interest yellow रे हमर gene of interest बाहर कला परे हमें एटा vector this is a vector this entire violet one is a vector एटा हमें जो एंजाइम रे कट करछे एटा GAATTC एटा भी हमरे जीन टाको भी हमें क्रोमोसोम भितरु GAATTC एक्जेक्टली सेम रेस्ट्रिक्शन एंजाइम रे ही कट करछे जोटा कि हमें प्लाज्मिड टाको भी कट करछे सेटा हेले ही हमर लाइगेशन नै परबो कॉम्प्लीमेंटरी एंड एठी देखंतु एटा हमर प्लाज्मिड कट हे गला तार TTAA फ्री एंड रहिला तळे TTA ए TTA एटा यार AATT सहित एनिल कर परिबो एनिल कर पारि कि एटा हमको गोटे रिकॉम्बिनेंट डीएनए हमको मिली पारिबो किंतु जदि से आउ को भी अनवांटेड साइड रे कट कला हमें एई रिकॉम्बिनेंट डीएनए हमें पाइबानी कारण हम स्टिकी एंड एनिलिंग कॉम्प्लीमेंटरी स्टिकी एंड हमको मिलिबानी आउ हमरा एनिलिंग हे पारिबानी then restriction enzyme that are used in construction of recombinant DNA are endonucleases which cause the DNA a specific recognition sequence. What would be the disadvantage if they don't cut? This is exactly the same one kintu alaga pattern re eta lekha heichi so we will be writing the same answer as we have written it over here. Then explain the significance of palindromic nucleotide sequence in the formation of recombinant DNA. So palindromic sequence हमको केमिति हेल्प करे palindromic sequence जो कट हैला आम पई से स्टिकी एंड प्रोवाइड करे it provides complementary sticky ends which helps the gene of interest and the vector to anneal together and ligase can then ligate or create a phosphodiester bond thus forming a recombinant DNA. So सेइति पय हमर जदी हमर palindromic sequence नथिबा हमको complementary एंड हमर मिलबनी कहार कॉम्प्लीमेंटरी एंड जीन ऑफ इंटरेस्ट और वेक्टर भीतर हमको कॉम्प्लीमेंटरी एंड ता हमको मिलबो ना then explain this this has been done then with the help of a simple sketch so the action of restriction enzyme eco r1 so let me go to the answer this is 21 just a minute 21 let me open the answer file so these are the answers जो क्वेश्चन ओडा को सब्जेक्ट क्वेश्चन ओडा को जो अछि एगुडा को मु तारा आंसर एठी देई नै छी so एटा हला सिंपल स्केच केमिति रेस्ट्रिक्शन एंजाइम केमिति मु सेई मोर पावर पॉइंट रु ही मु नै छी so this is a simple sketch how this restriction enzyme act so easily हमर एठी किछि डिस्क्रिप्शन किछि करबा दरकार नै से स्केच पचाछि so ए स्केच ता and also the student must be encouraged to learn this sequence palindromic sequence mane rakhiba gote ki dita restriction enzyme ro nihati important then with the help this has been done a gene has been ligated to a plasmid vector to prepare a recombinant DNA during bacterial transformation an exonuclease was added to the tube accidentally how will it affect the next step in the experiment? माने से कहउचंदी हमर गोटे रिकॉम्बिनेंट डीएनए ऑलरेडी हे जायछि जदि हमर रिकॉम्बिनेंट डीएनए हे जायछि से रिकॉम्बिनेंट डीएनए रो स्ट्रक्चर ता कोन से रिकॉम्बिनेंट डीएनए रो स्ट्रक्चर ता इज लेट मी से okay से रिकॉम्बिनेंट डीएनए रो स्ट्रक्चर ता कोन इट इज प्लाज्मिड भितर रिकॉम्बिनेंट डीएनए ता अछि सो आमे जो रिकॉम्बिनेंट डीएनए पाइले सेटा होची सर्कुलर सो से माने कहउचबी जो वाइल रे किबा जो ट्यूब रे से माने एक्सपेरिमेंट करछथि एक्सीडेंटली एक्सोन्यूक्लियस पड़ी गला एक्सोन्यूक्लियस माने कोन से डीएनए किबा आरएनए टा को नष्ट कर दला कोठो नष्ट करबो एक्सोन्यूक्लियस आमे डील करथिले एक्सोन्यूक्लियस ता सदा बेले बाहरु क्लीव करबा पई गोटे डीएनए टा ओपन थिबा दरकार लिनियर थिबा दरकार but over here the DNA is closed it is not linear तो DNA पाई तारो बाहरु ऐड करबा माने ओपन एंड किछि नै जोटु कि से ऐड कर परिबो कि एक्सोन्यूक्लियस कैन नॉट एक्ट ऑन अ सर्कुलर डीएनए जदि हमर गोटे रिकॉम्बिनेंट डीएनए हे जाईछि तो रिकॉम्बिनेंट डीएनए टा प्लाज्मिड यूजुअली वेक्टर उडा का हमर सर्कुलर थाए तो सर्कुलर जते बेले हे जाय था हमें एक्सोन्यूक्लियस जते बेले पकोउचे किछि भी तारो हबनी बट एंडोन्यूक्लियस हम जदि बाय चांस पड़ी जाउची तो से डीएनए टा को कट करके एंडोन्यूक्लियस क्लीव कर दे परिबो किंतु एक्सोन्यूक्लियस केबे भी क्लीव करि परिबो नाही then what is a vector list the properties of vector again we have to define what is a vector of properties oda ko ethi mo answer to mu deichi but over here also I have in the PowerPoint also it is present so what are the properties properties bhi dei paranti characteristic bhi dei paranti size vector 2 should be small in size jete choto haba easily isolate he paribo it must have a origin of replication that will enable them to replicate themselves tankaro origin of replication eti dekhantu ori sequence achi au jo vector re ori sequence na thae amara आमे आर्टिफिशियली आमे से वेक्टर रे ओरी सीक्वेंस को आमे ऐड करी थाउ सो दैट से गोटे गुड वेक्टर भलो वेक्टर हे पारिबो देन इट शुड हैव क्लोनिंग साइट्स माने कोन गोटे स्ट्रेच ऑफ डीएनए आमे ऐड करू सब वेक्टर रे क्लोनिंग साइट न थाए आमे जो क्लोनिंग साइट दरकार न थाए आमे आर्टिफिशियली सेटा तयारी करथाउ कोन क्लोनिंग साइट गोटे स्ट्रेच ऑफ ए रेस्ट्रिक्शन साइट आमे ऐड करू आमे गोटे एरिया रे हमर जदी इक्वार1 एससीए1 स्मा1 केपीएन1 स्मा1 हिन3 सब गोटे जगह रे माने रेस्ट्रिक्शन साइट उडा को जदी ए वेक्टर भीतर अछि सो आमे डिपेंडिंग मो पाखरे को रेस्ट्रिक्शन एंडोन्यूक्लियस अछि रेस्ट्रिक्शन एंजाइम उडा को बहुत कष्ट आमा पाखरे मो पाखरे सब तक रेस्ट्रिक्शन एंजाइम गोटे लैब भीतर सदा बेले रही परिबनी मो किंतु कॉमन रेस्ट्रिक्शन एंजाइम जेमिति कि इक्वार1 सब प्राय थाए केपीएन1 प्राय थाए हिन3 प्राय थाए सो आमे काहे कि अधिक आमे रेस्ट्रिक्शन साइट एटा आमे आर्टिफिशियली इंट्रोड्यूस करूछे आर्टिफिशियली माने ए जो देइछे आमे जो रेस्ट्रिक्शन साइट उडा को एठी देखंतु हिन3 रो AAGCTT एटा तापरे किछिटा सीक्वेंस आमे देई आमे सिंथेटिक डीएनए गोटे तयारी करथाउ जोतरे कि ए सीक्वेंस रा रहला तापरे किछिटा बेस पर चार्ट देई कि पीएसटी रा सीक्वेंस रहला तापरे इक्वार1 रा सीक्वेंस रहला तो आमे गोटे सिंथेटिकली आमे से डीएनए टा तयारी करी कि आमे तापरे ए रेस्ट्रिक्शन बा क्लोनिंग साइट डीएनए रो क्लोनिंग साइट रे एठी ए क्लोनिंग साइट रे आमे इंट्रोड्यूस करथाउ काहे कि हम इंट्रोड्यूस करू कारण मु जदी मोर जीन ऑफ इंटरेस्ट को इको आर1 रे काटिची तो एठी भी मु इको आर1 रे मोर वेक्टर टा को काटिबी मो वेक्टर टा को काटिबा दरकार सो वेक्टर टा को कोथरे मु काटिबी मते जानिबा भी मते दरकार मु सपोज हिन3 रे काटची मु एडा इको आर1 रे काटि देबी मोर केबे भी रिकॉम्बिनेंट डीएनए किबा क्लोनिंग हे पारिबो नाही सेति पय आम पाखरे ए क्लोनिंग साइट्स निहाती रहइबा दरकार then recombinant vector can be introduced into a host amara recombinant vector he sala pare se gote host bhitru ko nihati diba darkar a vector ta non toxic heba darkar a most important ta pakre gote selectable marker heba diba darkar selectable marker what is a selectable marker it helps in the detection of host which are having recombinant plasmid से किबा हमें सेटा को जो होस्ट भितरे रिकॉम्बिनेंट प्लाज्मिड थाए हमें ताको ट्रांसफॉर्म होस्ट भी कहि था काना से ट्रांसफॉर्म ही जाई छि ता पाखरे ओडे रिकॉम्बिनेंट प्लाज्मिड अछि सो हमें ट्रांसफॉर्म होस्ट सुडा को सिलेक्ट करिवार एबिलिटी का पाखरे थाए ए रिकॉम्बिनेंट प्लाज्मिड उडा का पाखरे थाए ओके सो जेमिति कि सेमानक भितरे एठी देखंतु एम्पिसिलिन रेजिस्टेंट जीन अछि सो मु जदी ग्रो करबी गोटे एम्पिसिलिन प्लेट माने हमर पेट्री प्लेट एठी देखंतु एटा होची गोटे पेट्री प्लेट आपण जो स्लाइड येलोइश घळिया जो बैकग्राउंड जो देखाउची सेटा होची मीडिया सेटा होची सॉलिड मीडिया ता भितरे बैक्टीरिया ग्रो करबा पे हमें सब किछि हमें पकेई थाउ हला सो एटा सपोज एम्पिसिलिन रेजिस्टेंट प्लेट जहा भितरे एई प्लाज्मिड ता पसिची एटी एम्पिसिलिन रेजिस्टेंट जीन खाली ए रिकॉम्बिनेंट डीएनए पाखरे अछि जो होस्ट बा जो बैक्टीरिया एउडा कहउचंदी बैक्टीरिया जहा भितरे एटी देखंतु तीनीटा ए जो दिया अछि या भितरे ही बैक्टीरिया भितरे ही हमर ट्रांसफॉर्म बा रिकॉम्बिनेंट डीएनए अछि सो एमाने ही ट्रांसफॉर्म बाकी का पाखरे भी रिकॉम्बिनेंट डीएनए नाही तो सेमानक पाखरे एम्पिसिलिन रेजिस्टेंट जीन नाही कि एम्पिसिलिन हमें जदी पकोउचे एम्पिसिलिन को नलिफाई करिवार सेमानक पाखरे एबिलिटी नाही सो सेमाने एम्पिसिलिन रेजिस्टेंट प्लेट रे मरि जिवे खाली जो बैक्टीरिया जो मानक पाखरे वेक्टर अछि सेमाने ग्रो करिवे एठी हमें जो देखुचे खाली एम्पिसिलिन रेजिस्टेंट बैक्टीरियल कॉलोनी उडा को ग्रो करचंती हमें एथरु नै कि हमें फर्दर हमें स्क्रीनिंग करू एक्चुअली अछि कि नै हमें स्क्रीनिंग करू सो सिलेक्टेबल मार्कर निहाती रहबा दरकार सो दीज आर द characteristic of features that must be present in a vector. Then differentiate cloning and expression vector. हमारा cloning vector और expression vector हमें एक खाली हमें ये भी कर दे परिवा define करके भी हमें differentiate कर दे परिवा हमारा types of clone over here. So cloning vector कौन है? A cloning vector, a small piece of DNA which are used to introduce the gene of interest into a host. से माने खाली gene of introduce टाको host भीतरे introduce करिवे किंतु protein produce करिवे ना करिवे आमे कहि परिवानी खाली हमको जेते बेले दरकार जे गोटे पीस ऑफ डीएनए टा हमें गोटे होस्ट भीतर को हमें इंट्रोड्यूस करबा ताहाले हमें क्लोनिंग वेक्टर यूज करबा बट हमर जेते बेले प्रोटीन एक्सप्रेशन हेबा दरकार हमको जेते बेले दरकार जे नाही हमर प्रोटीन टा से डीएनए टा जो हमें इंसर्ट करछे से जीन टा इट मस्ट बी एक्सप्रेसिंग तो हमें सेते बेले सदा बेले एक्सप्रेशन वेक्टर हमें यूज करबा लेट मी शो यू. So expression vector is a vector which not only introduces a gene of interest into the host but also aids in the analysis of the foreign gene by relevant protein product expression. माने से foreign gene टा जो आसिला से foreign gene रो एठी protein production भी एठी देखंतु ए जो green color रो is a foreign gene. It must be this vector must be having the ability to produce mRNA, that is messenger RNA from this gene, and also from the messenger RNA it must code the protein of interest. So this is the basic difference. हमें define करिति हमें तारो difference टा हमें कहि नहीं पारिबा से. Then what is the role of ori in any plasmid? Ori माने origin of replication. सेटा replication पाई निहाती दरकार. आमरो कॉपी नंबर मध्य आमरो डिपेंड करे आमरो ओरिजिन ऑफ ओरी सीक्वेंस ऊपर आमरो कॉपी नंबर ता भी आमरो डिपेंड करे. तापर व्हाट इज अ सिलेक्टेबल मार्कर? What is a selectable marker? हमें just हमें selectable marker हमें discuss करते हैं. Then glycosidase enzyme is considered as a better selectable marker justify the sentence. हमें एबे जो discuss करते हैं हमें गोटे ampicillin resistant selectable marker हमें discuss करते हैं. Now let us see से माने कहिचंदी glycosidase enzyme. Okay. So glycosidase enzyme ta amara ei blue green amara blue green colony produce karibare help karanti. Amara bacteria re lag jet gene achi. So se lag jet gene functional enzyme produce करे. Black jet gene functional enzyme produce करे जे कि in the presence of S girl गोटे substrate से गोटे blue coloration टे produce करिवो हला सो आमे जदी हमर लैग जेट जीन अछि आमे जदी लैग जेट जीन भितरे हमर जीन ऑफ इंटरेस्ट एटा आमे जो देखुचे व्हाइट कलर आमे जीन ऑफ इंटरेस्ट को इंट्रोड्यूस करते हमर लैग जेट रो सीक्वेंस भितरे हमर जदी फॉरेन ट्रांसजीन टा को इंट्रोड्यूस करचे माने से लैग जीन टा फंक्शनल नाही so से blue coloration of produce कर परियो so directly आमे एक color change हो आमे एक color change हो जो गुडा को blue से मानन को पाखरे vector नाही जो गुडा को white से मानन को पाखरे vector अछि so आमे directly so this galactosidase this lag jet gene produces your functional enzyme which is your beta galactosidase okay so this beta galactosidase metabolizes lactose to form your glucose and galactose glucose और galactose एटा ये करे so this S gal को भी से beta galactosidase gene can hydrolyze X gal हमें सदा बेले एक्सपेरिमेंट रे हमें लैक्टोज नहीं न खाओ हमें X gal नहीं खाओ S gal टा कोन करे गोटे ब्लू पिगमेंट कर दिए सो से ब्लू पिगमेंट जदी हेची ब्लू पिगमेंट हेची माने हमर इंसर्ट जीन ऑफ इंटरेस्ट हमर नाही प्लाज्मिड भितरे नाही किंतु ब्लू पिगमेंट जदी होउनी व्हाइट कॉलोनी ही फॉर्म होची ए व्हाइट कॉलोनी उडा का भितरे हमर जीन हमें देखु देखु हमें कहि देले जे these colonies are carrying the gene of interest that's why it is a much better selectable marker then while cloning vector which of the two will be preferred by biotechnologists bacteriophage or your plasmid आमे क्लोनिंग कला बेले बैक्टीरियोफाज आमे यूज करबा किंबा प्लाज्मिड यूज करबा जस्टिफाई द रीजन आमे सदा बेले बायोटेक्नोलॉजिस्ट बैक्टीरियोफाज यूज करिवे कारण बैक्टीरिया फॉर क्लोनिंग ऑफ अ प्लाज्मिड कारण बैक्टीरियोफाज रो कॉपी नंबर बहुत अधिका सेटा बहुत बड़ा इंसर्ट suppose गोटे एग्जांपल नोची नॉट ने suppose गोटे प्लाज्मिड 1 kb और 10 kb पर्यंत नै पारिबो से 10 kb पर्यंत गोटे डीएनए कैरी करि पारिबो बा ट्रांसफर कर पारिबो बैक्टीरियोफाज 10 kb 10 किलो बेस रु अधिका से डीएनए ट्रांसफर करि पारिबो सो ए सब फीचर्स उडा को नै कि हमर मामे प्राय एनी साइंटिस्ट विल बी प्रिफरिंग दिस बैक्टीरियोफाज ओवर प्लाज्मिड यार भी आंसर मु आंसर सेक्शन रे मु देई नै छी so what are the features of the plasmid being used for cloning again से जो tools हमें जो ये करथिले तार ही repetition so we can write this down write a note on cloning vector लेखि कि हमरो cloning vector एठी मु देई नै छी cloning vector कोन so cloning vector लेखि सारी की this type of cloning vector plasmid cosmid phase artificial chromosome again हमरो नोट जो दिया आइची हमरो एटा शॉर्ट नोट दिया आइची बेले बेले लॉन्ग क्वेश्चन भी पचार दियंती क्लोन वेक्टर्स ऊपर जदी लॉन्ग क्वेश्चन पचाले हमको क्लोनिंग वेक्टर एक्सप्रेशन वेक्टर दीटा जाको डील करबा पई पड़िबो खाली जदी क्लोनिंग वेक्टर लॉन्ग क्वेश्चन जदी दिया आइची वी हैव टू डिफाइन इट we have to also cloning vector ro features उडा को कोन अछि तार वेक्टर हेबार features उडा को कोन एई गुडा कोन एई जो features उडा को हम जे डील कले एई गुडा कहीं सेकरे पुनी लेखिबा को पड़िबो लेखि साला पर टाइप्स ऑफ क्लोनिंग वेक्टर हमको देबा को पड़िबो लॉन्ग क्वेश्चन जदी अछि शॉर्ट क्वेश्चन जदी अछि हम एतक फीचर लेखि कि टाइप्स भी लेखि कि गोटे डायग्राम सिंस मु गुडाई जगह रे डायग्राम देनी मोर आंसर उडा करे सो students should be encouraged being students of biology they should be encouraged always to draw a diagram गोटे simple cloning vector रो से माने डायग्राम भी दे देले भी माने it is more attractive and more mark fetching then name the various cloning vectors and how a plasmid can be used as use for genetic engineering आमर एटी various cloning vector आमे गुडाय cloning vector रो जो अछि आमे सेई जाको आगो vector एटी आमे define करिबा तापरे various cloning vector आमे define करिबा तापरे आमे directly plasmid को दिबा plasmid कोन सेटा किमिति हमरो cloning रे use हे परिबो it can be again हमरो tools आमे जदी छुआ माने जदी पूरा मने रखि देई पारंती tools जदी मने रखि देले तो केमिति सेटा cloning रे use कर परिबे आमे आराम से सेथुरु ही नै जिबा so it is having a origin of it should be having your restriction site माने cloning site जदी अछि सेठी restriction site उडा को अछि आमे easily cut कर नै परिबा so after ligation it can be introduced this plasmid are easily introduced into host after introduction into host yes it is having a origin of replication say host bitare independently autonomously replicate kari paribo so because of these features it can be used for cloning purpose cloning vector hela eta then explain importance of OREA ampicillin R rope so this is 3480 already we have discussed most of the things but rope thing is rope one is new one so this rope ampicillin R होची हमर रेजिस्टेंस हमर सिलेक्शन रे हेल्प करे ओरी तो हमें ऑलरेडी जान सके हमर रेप्लिकेशन रे हेल्प करे ऑटोनोमस रेप्लिकेशन रे हेल्प करे और कॉपी नंबर multiple copy number produce करिबारे भी हेल्प करे rope is goes for the protein involved in replication of plasmid और replication ପାଇଁ गोटे जो प्रोटीन दरकार सेटा ए rope sequence सेटा कोड करे then this 35 what is a cDNA how they are synthesized we sorry what is a how they are synthesized ame already eta ame cDNA re ame discuss kar sache cDNA library what is cDNA library how they are constructed so this is also present over here and the answer have been provided I am hurrying I am sorry because I have to complete many things so this is let me show so this is a cDNA library ame khali definition ame dei dele cDNA library ro so it is a collection of cDNA clones generated in vitro from functional mRNA of single tissue or cell of an organism with the help of reverse transcriptase stored in bacteria so what we are doing is ame gote suppose at a particular moment ame liver cells nele ba liver ro godi chotiya खंड नेबा so से समय रे तार से cells उडा का liver cells उडा भितरे जेति के mRNA थिबो mRNA messenger RNA आ किछि भी RNA ना mRNA आमे से उडा को isolate करछे सब mRNA रो हमरे cDNA construct करछे cDNA construct कला का परे we are introducing it into vector सब तक cDNA आमे vector रे introduce करछे vector रे introduce कर साला परे those vectors we are introducing into any host हला तो प्रैक्टिकली कोन हमर सेल भितरे जेते mRNA माने कोन फंक्शन से माने जीन रु mRNA हमर आसीची तो बेसिकली हमर जेते फंक्शनल जीन अछि तार mRNA हमें जदी नेचे cDNA करछे अगेन हमें से फंक्शनल जीन उडा को विदाउट इंट्रॉन इफ इट इज अ यूकैरियोटिक जीन हमें से जाको विदाउट इंट्रॉन रे sorry eukaryotic mRNA हमें without intron रे हमें से जाको तयारी करे exactly मु एटा जोटा आगुरु डिस्कस करथिबी so without intron मु एटा ए mRNA टा नुके नै कि मु जदी cDNA करछि मोर without intron रे मोर फंक्शनल eukaryotic फंक्शनल जीन मिलला आ प्रोकैरियोट रे मु जदी mRNA करूछि प्रोकैरियोट रे तो नॉन फंक्शनल माने स्ट्रक्चरल जीन टा पूरा कंप्लीट तार इंट्रॉन नाही हमरे डायरेक्टली mRNA को नै कि हमें cDNA करने परिबा सो हमर जेतक जीन अछि प्रैक्टिकली जेतक जीन से समय रे एक्सप्रेस होउछि काना सब जीन सब समय रे एक्सप्रेस होतबा माने नाही जेतक जीन से समय रे एक्सप्रेस होउछि हमें से जीन रो डीएनए उडा को नै कि हमें गोटे लाइब्रेरी तैयारी कर देले हमें ओडे लाइब्रेरी तैयार कर देले आगु को हमकु जहां भी दरकार हबा हमें से लाइब्रेरी उडा करू लाइब्रेरी माने से cDNA गुड़ाक प्लाज्मिड अच्छे प्लाज्मिड गुड़ाक इंट्रोड्यूस हो जाए ई कॉली को आउ ये ई कॉली गुड़ाक आम प्रिजर्व कर नौचे -70 रे कि लिक्विड नाइट्रोजन लेबल करके हमको जेते बेले सपोज मते इंसुलिन cDNA दरकार मु लेबल करके रखिथिबी इंसुलिन cDNA तो मु मोर लाइब्रेरी रो इंसुलिन cDNA मु बाहर करके मु यूज कर पारिबी सो दिस इज द cDNA library. Then in recombinant DNA technology, what do we mean by host? So let us come to a host. A host are living system or cells which are carrier of recombinant DNA molecules or vector and can be propagated or multiplied and also are known as the cloning factories. They are different type of host, your prokaryotic host and your eukaryotic host. Jadi short re palla. तापर हमको host रो characteristic features भी next पचाथिबे. So, what are the properties of your host that enables them to be used for recombinant DNA technology? 80 properties of host ki amara ta pare pare achi. So these are the properties of host or characteristic host. Rapid turnover, fast growth, then can be grown in large quantities, non-pathogenic, then capable of accepting plasmid or bacteria, maintain foreign gene through multiple generation. से माने divide करिकी माने daughter cell मान को से माने जैसे बड़े divide करंती daughter cell मान को भी से माने ये जो recombinant DNA अच्छे recombinant DNA टा को transfer करिबा दरकार नहले हमारा से ही host टा को कामरा आसीबनी. Then will secrete a high yield of protein from the expressed foreign gene. से माने final product which is protein से इटा को भी से माने express करिबा दरकार. So these are the characteristics of the host. Then what are competences? What does the word competence refers to? How can we make the host competent? ए सबु गुडाक हमरो competency ऊपर हमरो ए सबु गुडाक क्वेश्चन गुडाक बेस्ड अछि. So let me go to the answer. Just a moment. So exactly मु जो गुडाक मु ये करछि तार आंसर गुडाक भी दे देछि. Competent cells. So competent cells are those that allow foreign DNA to be incorporated into the host by slight alteration in the cell wall. Normally आमे खाली जदी E. coli नेबा एठी देखंतु एटा होछि E. coli आमे जदी खाली E. coli नेबा खाली E. coli नै कि आमे जदी recombinant DNA टा को इंट्रोड्यूस करबा मीडियम रे माने बाहर तार वॉल उडा को केबे भी से प्लाज्मिड टा नेबेनी बा recombinant DNA टा को नेबेनी आमे तार वॉल टा को मॉडिफाई करके किमिति मॉडिफाई करके आमे एमिति मॉडिफाई करके जे ता वॉल रे पोर्स ट्रांजिएंट पोर्स टेम्पररी पोर्स क्रिएट होची से टेम्पररी पोर क्रिएट हला माने से पोर थ्रू रु हमर डीएनए टा एंटर करिवो आमे टेम्पररी पोर केमिति क्रिएट कर परिबा आमे कैल्शियम क्लोराइड आमे देकी आमे टेम्पररी पोर्स क्रिएट कर परिबा मु एजा को सब लेखि नै छी आंसर रे मु एजा को सब लेखि नै छी आमे ये करिकी सॉरी आमे कैल्शियम क्लोराइड ट्रीटमेंट देकी आमे टेम्पररी पोर्स आमे इंट्रोड्यूस कर परिबा किबा आमे इलेक्ट्रिकल चार्ज देकी भी आमे कॉम्पिटेंट आमे ए टेम्पररी पोर्स यूज कर परिबा किबा आमे फ्रीज एंड थॉ करिकी भी आमे ए टेम्पररी पोर्स यूज कर परिबा. सो आमे आइदर केमिकल कैल्शियम क्लोराइड किबा फ्रीज एंड थॉ किबा हीट ट्रीटमेंट देला परे ही गोटे सेल कॉम्पिटेंट हुए. कॉम्पिटेंट माने गोटे रिकॉम्बिनेंट डीएनए टा बाहरु ता निज भितर को नेबा पे से कॉम्पिटेंट हेई थाए. Okay. Then what do you understand by gene cloning or recombinant DNA technology? एटा हम recombinant DNA technology जो स्टेप गुडा को जो अछि एठी से स्टेप गुडा को हमको ऑलरेडी तार ओवरव्यू मु कहि सालेनी. सो से स्टेप गुडा को हमको अगेन देबा पे पड़िबो. नेम एनी टेक्निक by which DNA fragment can be separated. DNA fragment gel electrophoresis द्वारा सेपरेट. एटा एक्चुअली दीटा अलग अलग क्वेश्चन किनु मु एकाठी दे देछि. Explain the principle of separation of DNA fragment in gel electrophoresis. So आमे gel electrophoresis रे आमे separate करी नहीं परिबा. So how is this? एटा केमिति competent करी परिबा तारो एटा sorry not competent एटा DNA cloning टा केमिति हबो एटा तारो process टा. So gel electrophoresis से आमे DNA किंबा RNA आमे आराम से आमे DNA RNA प्रोटीन आमे आराम से तांको साइज हिसाब से आमे सेपरेट करछे. जो मानक छोटो साइज से माने आग रन करिवे जेल भितरे आ जो मानक बड़ साइज से माने तांकर मूवमेंट रे से माने स्लो आमे जेमिति जणे टिके माने ओवरवेट ओबेज अछंती से माने चालिला बेले से माने टिके कोन आस्ते चलंती किंतु जो माने फिट अछंती से माने बहुत जल्दी आगुआ चली जाइतबे एक्जेक्टली जो मानकर साइज डीएनए आरएनए किबा प्रोटीन रो यार छोटो साइज थिबो से माने ए जेल थ्रू रु इजीली किंतु जेल रे माने चालंती मु जो कहे जेल थ्रू रु जो माइग्रेट करंती केमिति माइग्रेट करंती आमे गोटे इलेक्ट्रिक फील्ड आमे देई थाओ. ओके पॉजिटिवली चार्ज मॉलिक्यूल मूव टुवर्ड्स द कैथोड and negatively charged molecule move towards the anode, vice versa. Our DNA is negatively charged, so negatively charged DNA ta anode towards anode को से move करिवो. So ଆମର ए जो migration टा हुए DNA RNA किबा protein it is dependent on the electric field. So ଆମର जेल भितरे आमे DNA टा को देखिबार हमर स्कोप ने आमे नॉर्मली जदी इलेक्ट्रोफोरेसिस जेल आमे जदी देखुचे आमे ता भितरे आमे DNA टा को देखि परिबानी. तो DNA टा को जेल रे विजुअलाइज करिबा पे नेक्स्ट क्वेश्चन टा दिया है कि हाउ वी कैन विजुअलाइज आमे इथिडियम ब्रोमाइड आमे पकई थाओ. Okay, this intercalates with the DNA and gives orange band on exposure to ultraviolet light. आमर जेते बेले आमे से जेल टा को आमर रन हे गला आमे इलेक्ट्रिक करंट आमे यूजुअली डीएनए टा को लोड कर साला परे आमे गोटे इलेक्ट्रिक करंट अप्लाई करू यूजुअली 40 to 50 वोल्ट रे आमे सेटा को रन करू. 40 to 50 वोल्ट रे अबाउट 45 मिनट टू 1 आवर रे रन कला परे आमे से जेल टा को बाहर करी नै कि आमे यूवी लाइट ऊपर आमे रखि थाओ. आमर गोटे बेस थाए आमे जेमिति एक्सरे आमे जेमिति देखि थाओ हॉस्पिटल रे गोटे पछपटे लाइट थाए से लाइट टा नॉर्मल लाइट सेमिति का हमर गोटे बेस थाए जो थरु कि यूवी ट्यूब गुडा को लागि थाए आमे जेते बेले से जेल टा को रखू जोटी डीएनए थाए से ग्लो करिवो काहे कि ग्लो करिवो कारण सेठी इथिडियम ब्रोमाइड से डीएनए सहित लागि जाईची किंतु ए इथिडियम ब्रोमाइड टा हाईली कार्सिनोजेनिक व्हाइल डीलिंग विथ दिस इथिडियम ब्रोमाइड आमे प्रॉपर गियर्स ने कि ग्लव एंड एवरीथिंग हमें पिंदी की यूज करबा दरकार एप्रॉन सब पिंदी की यूज करबा दरकार आ अल्ट्रावायलेट लाइट भी हमरो आखि पाई आम पाई बहुत खराब. So usually we wear a spec which is ultraviolet protected and also the UV light is also inside a shield. UV लाइट टा भी गोटे शील्ड भितर ही थाए ताकि आम ऊपर को डायरेक्ट रेडिएशन टा UV लाइट टा आम ऊपर को न आसे. Now let us go to the next question. So I have already dealt with how can we view a mixture of fragmented DNA was running on agarose gel. The gel was stained with ethidium bromide but no band was observed. What was the cause? So एमिति भी हुए बेले बेले हमर बैंड हमको देखा जाए माने हमें नुआ नुआ जेते बेले हमें एक्सपेरिमेंट करथाओ हमको बैंड देखा जाए. सो तारा आंसर कोन बेले बेले कोन हे थाए हमें कैथोड एनोड हमें रिवर्स ओरिएंटेशन रे हमें कर दे थाओ. हमें एनोड और कैथोड टा को रिवर्स ओरिएंटेशन रे हमें कर देले भी हमर हमको देखा जीवनी माने से बाहर की से माइग्रेट है कि हमर बफर भीतर को पळेबा हमर देखा जीवनी. डीएनए टा बहुत अधिका जदी डाइजेस्ट हो जाइथिबो भी हमको देखा जीवनी. Then electrode are placed in opposite ए जो रिवर्स ओरिएंटेशन मु जो कहली इलेक्ट्रोड्स आर placed in opposite direction in the gel assembly ताहाले भी हमको देखा जीवनी. The DNA moves out of the gel and we are unable to see the GNA. Then maybe sometimes even जदि हमें इथिडियम ब्रोमाइड ठीक अमाउंट रे भी जदि हमें पकेनी ताहाले भी हमको देखा जाई पारिबो नाही. Then how is recombinant DNA transferred to host? There are various mechanisms of transferring the recombinant DNA into host. मु तो लेखी मो आंसर लेखी देछि तथापि I just wanted to like it might be transformation, transfection, electroporation, microinjection, bioelastic liquefaction. So depending upon the mark जदि खाली 2 मार्क अच्छी आमे खाली ना लिखिदेले 3-4 मार्क अच्छी हमको सबुथर डेफिनेशन देबाको पड़िबो लॉन्ग क्वेश्चन जदि अच्छी वी नीड टू give the diagram over there also diagram देखि कि हमको सब तक डील करबा को पड़िबो then why DNA cannot pass through cell membrane how can a bacteria be made competent एटा already हमें डील कर साले कारण आमर बैक्टीरियल वॉल जो अच्छी आम बैक्टीरियल वॉल रे माने एमिति किछि गैप नाही जहा थ्रू रु कि डीएनए टा एंटर कर परिबो आल्सो वी नो दैट द प्लाज्मा मेम्ब्रेन इज अ लिपोप्रोटीन थ्रू रु चार्ज प्रैक्टिकल पार्टिकल्स उडा का आमर लिपिड अच्छी आमर तो चार्ज पार्टिकल उडा को केबे भी पास करि पाइबो नाही plasma membrane through so because of this reason what we need to do is we need to make a cell competent ame competent केमिति करथिले calcium chloride दे कि electric charge दे कि किंबा फ्यूजन थ करके हमें competent करछे ताकि पोर ट्रांसिएंट पोर माने परमानेंट नहीं ट्रांसिएंटली गोटे पोर क्रिएट हे कि संगे संगे से पोरटा बंद हो जिवो आ इट इज अ हिट एंड ट्रायल मेथड से जो ओपन हला से ओपन हेबा भितरे जेति कि एंटर करछथि से एंटर करछंती बाकी आउ एंटर बाकी रिकॉम्बिनेंट डीएनए आउ एंटर कर परिबे सेति पय हमर ट्रांसफेक्शन किबा ट्रांसफॉर्मेशन एफिशिएंसी ता बहुत कम आमे जदी 1000 टा नेथिबा अराउंड 7-8 टा भितरु को हमर व्हाट इज इट रिकॉम्बिनेंट डीएनए टा एंटर करिथिबो देन एक्सप्लेन एनी थ्री मेथड ऑफ वेक्टरलेस ट्रांसफर ए भितरु वेक्टरलेस ट्रांसफर माने हमको जो थरे वेक्टर दरकार नहीं होची माइक्रो इंजेक्शन रे हमको वेक्टर दरकार नहीं कारण हमें डायरेक्टली डीएनए टा को न्यूक्लियस भीतर को इंजेक्ट करछे बायोलिस्टिक वी आर यूजिंग जीन गन सो एथिरे भी वेक्टर दरकार नहीं हमें डायरेक्टली डीएनए टा को हमें सेल्स ऊपर को हमें बोम्बार्ड करछे एंड लिपोफेक्शन रे भी हमरो वेक्टर दरकार नहीं हमें लिपोफेक्शन रे लिपिड लिपोसोम्स यूज करके हमें इंट्रोड्यूस करूछे So these are the three methods. Then coming to the next one, what is PCR? List the three main steps. 18 mu सबुतक क्वेश्चन एकाठी गोटे लॉन्ग क्वेश्चन रे दे देछि जदी शॉर्ट क्वेश्चन आसे खाली व्हाट इज PCR आसी पारे लिस्ट द मेन स्टेप्स इन PCR आसी पारे सो द स्कीमेटिक डायग्राम भी हमको पचारि पारंती एप्लीकेशन भी पचारि पारंती So I have written the answer over here also. The answer has been given. Let me just see. It's the question number 49. So the definition of the PCR has been given. Here discover करथिले कैरिमुलिस so हमको टाइम मिल नथिला एमसीक्यू रे हमें पीसीआर जाइने so पीसीआर किए फर्स्ट को साइंटिस्ट पीसीआर ये करथिले डिस्कवर करथिले इट इज कैरिमुलिस इन 1984 so तार तीनटा स्टेप अछि denaturation annealing and extension and these are the application of PCR diagnosis of pathogen हम बॉडी एबे हमें PCR जो RT-PCR कहूचे RT-PCR रिवर्स ट्रांसक्रिप्टेज PCR रिवर्स ट्रांसक्रिप्टेज PCR माने कोरोना वायरस इज अ आरएनए वायरस हम बॉडी हमें RT-PCR जो कहूचे से आरएनए वायरस आरएनए टा को नै कि हमें रिवर्स ट्रांसक्रिप्शन एग्जाम दे कि हमें डीएनए अधिक तैयारी करछे हमें जे डीएनए तैयारी करके हमें चेक करछे जे एक्चुअली से जो डीएनए टा कोरोना वायरस रो कि नाही Okay, so RT-PCR is based on PCR only. PCR टा कोना हमें गोटे DNA टू गोटे DNA रो हमें billions of copy of that DNA we can make by using a PCR machine. So we have already dealt with this PCR. मु खाली आउ टिके खाली से PCR machine टा को मु देखै नेबी. Just a moment. So this is a PCR machine. दिस इज ए मशीन टारे हमको जहाँ दरकार माने गोटे डीएनए को एम्प्लीफाई करबा पे पॉलीमरेज देन ऑल द डीएनटीपीस वी टेक इन दिस आपन को देखा जाउतबा गोटे वायल देखा जाउतबा ट्यूब गुडा ए ट्यूब गुडा को भीतर हमें सब नेइनो ए पीसीआर मशीन रे हमें रखिकी तापरे ए सब साइकिल तीनटा जाक साइकिल जहाँ भी हमको जेतिकी टेम्परेचर दरकार हमें यार एटा कंप्यूटर टा अछि सो एठी हमें फीड करिनो फीड आ आजिकाली एटा होची बेसिक पीसीआर मशीन आजिकाली पीसीआर मशीन उडाको से माने गोटे डेस्कटॉप को अटैच हेचंती डायरेक्टली जहाँ भी रिजल्ट बाहरला हमको से कंप्यूटर रे हमको मिली जाए. So am I having more time? Or?
No.
Okay. Okay, I'm sorry. So I'm having all the question over here, and also there are 80, there are 60 questions more, maybe 15 question I have not discussed. So I'm having the answers also over here. So please go through the answers also. If anyone is having any doubt, then just for a few minutes, I can clarify the doubts. I hope I will be able to clarify your doubts. किसी doubt तो अच्छी की? So you can also directly, if you are having any doubts also, you can also call me during the evening hours. My number is 94371. Again, 94371 76655. 94371 76655. So I'll be very happy to clarify any of your doubts. Thank you so much. Thank you so much for cooperating. And I'll be giving all the questions over here.
Thank you, madam.
Thank you,
madam.
Now we close the today's session, and when next biology turn comes, botany and zoology, that has been given to you. So you can see to that. And as madam has told, if you have got any doubts, you can contact madam in the evening hours after 7 or PM or something like that. If you have got any doubt, madam will be able to answer it. Thank you very much for your patient hearing and being with us for last 4 hours or more than 4 hours from 9 AM up to now. Thank you very much. Okay.
Thank you, sir.
Thank you, sir. Thank you, sir.